Okay, welcome to the Botanical Safety Consortium annual virtual meeting for 2020. I know some people are still connecting but wanted to go ahead and get started because we have a lot of information to get through today. I appreciate everybody joining virtually from wherever you might be. I'm Michelle Embry, the Associate Director of the Health and Environmental Sciences Institute, or HESI. We're the conveners of the consortium. And I'm just going to go through some very brief introductions and objectives before we launch into the program for today. So the objectives of the meeting are to provide basic information on the structure and the mission and objectives and strategy of the Botanical Safety Consortium, or BSC for short. We're going to share some brief high-level overviews of the current technical working groups, which are our scientific committees that are working to realize the objectives and strategy of the BSC. We're hoping with all of you who are participating today to be able to recruit some additional Botanical Safety Consortium stakeholders, and we'll talk a little bit about how you can become more involved in this project. And then towards the end, we're hoping to solicit some critical input from all of you into the scientific challenges and some of the opportunities that we face related to dietary ingredient safety of botanicals. So I'm just going to briefly walk through the agenda for today. So I'm Michelle Embry doing a brief introduction and objectives. Then we'll switch to Dan Marsman from Procter & Gamble, who will give us an overview of botanical safety, talking about some of the struggles and the needs and hopefully solutions that this consortium will address. Connie Mitchell, who's my colleague at HESI, will talk specifically about the public-private partnership and the organization of the Botanical Safety Consortium. Cynthia from the National Toxicology Program will give us an overview of the research on botanical safety within that program. Cara Welch will talk about FDA's dietary supplement program. And then we'll move into the more technical portion of the meeting today, where Joe Dever will talk about the technical working groups within the Botanical Safety Consortium, giving a high-level overview of their mission and objectives and the overall strategy. Stefan Gafner will talk more specifically about two of our other working groups that are focused on chemical characterization and data analysis. And then Jim Griffiths will talk and give us some conclusions and wrap up with some key questions that we would like you to provide input on. We will have about 45 minutes at the end for discussion and questions from all of you, and hopefully if all of your questions aren't answered on this virtual meeting today, there's plenty of opportunity to email any one of us, and we would be happy to get back to you. So just to cover some logistics for this virtual meeting, the entire meeting will be recorded and made available on our website, as well as PDFs of the slides. All of the participants on this, other than the speakers, are muted, and questions for either the individual speakers or towards the end of the meeting should be typed into the question box. I'll show a slide shot of that in a minute in your control panel for GoToWebinar. We're planning to have around 5 minutes or so after each of the individual presentations for clarifying questions. We ask that you hold the deeper dive, sort of high level questions about the consortium and the specific whether it's endpoints or botanicals or those sorts of things you'd like to talk about towards the end of the webinar in that 12 to 12:45 Eastern time slot, where we'll have much more time for discussion and additional questions. If you have any problems during the webinar, please email me, Michelle Embry. My email address is there, and it's also available on the Botanical Safety Consortium website, botanicalsafetyconsortium.org. So to ask questions, I have two screenshots here. Hopefully this is somewhat similar to what you see right now. We have the desktop view. For those of you who actually have GoToMeeting installed on your computer and you downloaded it, it should look something like this. And you just have to expand that questions tab in the control panel and type your question in, and we will see that. The panelists will see that. For those of you on the web through Instant Join, there is a question mark in the panel, and that's where you'd click on that, and that should pop up a window for you to type in your questions. So with that, I just wanted to put up here really quickly the website. my email, as well as my colleague Connie Mitchell's email, and our general email to the Botanical Safety account. I appreciate everybody joining in. I know more folks are going to join as this goes on, but with that, I'm going to turn it over to Dan Marsman, who is at Procter & Gamble, and he's going to kind of kick us off with an introduction to why we're working on this topic. So Dan.
Thank you, Michelle. And good morning, good afternoon, good evening, everyone. This is a pleasure. It's a pleasure to be with you. Welcome to the inaugural public meeting of the Botanical Safety Consortium. I'm Dan Marsman from P&G Healthcare, and I'm excited by this opportunity to share a little bit on the vision of the consortium, as well as to solicit your participation. In my introductory remarks, I'd like to give a brief overview of the science of botanicals and some of our current safety challenges, and then move into the conceptual development of the BSC. Well, it comes as probably no surprise to this audience that there is broad consumer demand for natural products, and in particular for the dietary or food supplements. Recent data suggests that over half of those dietary supplement users are also taking at least one botanical product. Many of those botanical products are sort of the tried and true botanicals that we are familiar with, but just in this list of the top 12, there are also the impression of increasing popularity of specialized or exotic botanicals. So certainly we are all exposed to botanicals in our everyday life. Dietary fruits and vegetables are as old as time. And so, but it's with the rise of the popularity of botanicals that we appreciate that realistically our ancestors had to learn that these are a highly diverse and creative array of plants. And among them, we recognize that there are really the good, the bad, and the ugly, from everything from dietary mushrooms and our beloved coffee beans to a variety of ingredients that are used to derive botanical drugs, such as taxus and salix and digitalis and poppy, but it also includes those that can have exquisite levels of toxins, such as ricinus or the death cap mushrooms. So certainly safety is a concern for botanicals. First and foremost, I can't help but mention adulteration. It's a particularly troubling area for both the public and the responsible industry. The unacceptable inclusion of intentional economic adulterations is a real problem, but the focus really for the discussion today is the general concerns that also exist with uncertainties about botanicals, and it's important to have that level of transparency about what kinds of constituents and the safety of those botanicals. Certainly, botanicals are complex. You'll hear me say that multiple times in this short presentation. But that complexity comes in multiple dimensions that I hope to illustrate in the next couple of slides. You know, it's recognized that there's over 20,000 plants that are thought to have medicinal benefits. And while many of these have been exploited for human use, very few have actually been fully characterized. When we talk about characterization, that characterization is realistically a challenge, or has been. And so for something as simple as a botanical such as clover, it still can contain hundreds of constituents. So it's a challenge to identify those constituents. But speaking as a toxicologist, it's important to create a realistic safety assessment. So it's not just about identifying each of those constituents, but being able to quantify them for a true risk assessment. When we look at that complexity, though, it's not just on the analytical front, it's honestly in the sourcing of those botanicals, all the way from our agricultural practices. We certainly want to advocate for good agricultural practice, but even with good agricultural practice, we clearly recognize that there are a variety of things that can modify the botanical, including the harvest conditions, so that in that context, when we harvest or where we're harvesting, like in an arid environment or a more fertile environment, can modify the botanical, and certainly how we harvest. The picture in the lower left illustrates an example of that, where the feedstock for one particular botanical clearly has a variety of components included in one botanical ingredient. In addition to agricultural practices, there's complexity that comes in the next stage as those botanicals are processed and put into a product. For a holistic evaluation, that complexity can result through the extraction processes of either expanding the complexity of the botanical or contracting that and concentrating components of that particular botanical. When we put all these things together, it's abundantly clear that there are a variety of ways in which a botanical ingredient can be complex and modified, and the introduction of things like contaminants or adulteration, or just the ordinary modification that occurs in the development of a botanical ingredient for a botanical product. But I would be remiss if I didn't also talk about exposure, because when we talk about botanical safety, the other half of that is a quality safety risk assessment. It only begins with the ingredient itself, but a full risk assessment needs to understand exposure, which can cover the full spectrum of concerns, from the dose to the subpopulation that might be targeted. Dietary supplements, you will notice, are sort of the focus of my opening remarks, but basic safety principles for botanicals apply regardless of the product form, including botanicals that would occur in cosmetics or personal care or specialty foods. All of that would have its application in a better integrity of the raw materials and understanding that exposure scenario. Well, how do we think about testing botanicals when we have a botanical and there is concerns? Traditionally, the testing schemes were built around single chemicals, and as you can see in this slide, toxicity testing and safety evaluation is a process that typically was catered towards single chemicals. We're thinking here about drugs or commodity chemicals or pesticides. In the case of drugs, we think of that in terms of a regulatory structure that is focused on ensuring safety and efficacy. And we assume that there may be harm until we can prove these ingredients to be safe. That they are typically isolated constituents and chemicals, and so they are simple and consistent, and the biological activity is focused on that simple chemistry. Botanicals are almost the polar opposite of that, right? They are a regulatory structure that, at least in the US, is aimed at ensuring access, and they're assumed to be safe until proven harmful. They are complex, as I've said numerous times already, and that biological activity is provided to the whole mixture, not necessarily to any one constituent. The important part of the botanical side of the equation is that assumption of safety. It is a hallmark of the botanicals because they are a natural part of our food chain. This is clearly something that we recognize, and we're thankful for the many efforts that others have gone to great extremes to establish already a solid foundation for safety for botanicals. I have here just a couple of publications to make that point, but it's clear that much work has already been done, especially on the international front, to progress the definitions of what a safe history of use is, and good agricultural practice, and good manufacturing practice. All of these things are critical, but particularly the safe history of use is an important part of establishing the safety of a botanical. And we're talking here about historical records that can establish the dose and the duration, the type of population in which this particular botanical has been used in the past, and what exactly is that botanical and how was it prepared and extracted and administered. So the question quickly becomes, as we see the enhanced interest in botanicals, is what if indeed the botanical is not identical to that history of use? Your botanical is of a different extract or even a different species, but very similar. Where do we go from there? Well, traditionally, that would be toxicology. For resolving these uncertainties, we would use traditional non-clinical testing. Unfortunately, traditional toxicology tools are generally not fit for purpose for botanicals. For a novel botanical, the current safety qualification process is highly dependent upon animal testing, but there's often a high degree of variability when we're evaluating mixtures. And what we recognize is that it's often an insensitive tool for discerning minor variations, like I was just describing, between raw materials. When you have all of those minor variations and they continue to occur over time, it really becomes problematic as a resource and a time-intensive process, and that notwithstanding the moral and ethical considerations of animal use. The good news is, fortunately, we have new methodologies that continue to be developed, including advances in chemical analysis, which provides a much clearer picture of the composition of the raw material and the variance between those raw materials. And comparative chromatography will give us both qualitative and quantitative assessment of the differences between raw materials. This is such a critical advance in the science of analytical chemistry, and therefore sets us up for the ability to apply that knowledge into a framework that indeed there has also been such progress on the toxicology side with current safety methods that involve both in vitro and in ex vivo and in silico tools. We have a variety of opportunities that provide real promise for the evaluation of the toxicologies of mixtures. These advances in characterization and safety testing are the fuel that actually brought together a diverse array of scientists, and that becomes the focus of the rest of the morning. Through a series of international meetings, first in Europe at Eurotox, and again in the US at the International Congress on the Science of Botanicals, there was an array of discussions talking about the opportunities to apply chemical characterization and new tools of toxicology to address the safety concerns associated with botanical mixtures. And this became an important conversation, and clearly It has grown to be quite a diverse array from both the public and the private sector in a broad conversation that continues even now. So with that, let me just summarize and say there's growing demand for botanical products and a growing trend towards modified or specialty botanicals. That diverse botanical starting material coupled with variable agriculture and processing practices has created challenges that we all face on both the public and private side. And therefore there's an ongoing need to bridge to various materials and have a transparent discussion about how one might qualify new botanical materials. And the good news is that there is truly opportunities in front of us to apply 21st century methods to address these concerns. Out of this common understanding, the concept of the Botanical Safety Public-Private Cooperative was born. What you're about to hear is the BSC's proposed path forward, driven by our work and our working mission statement, which is to enhance the botanical safety toolkit and bring clarity to botanical dietary ingredient assessments for both manufacturers and regulators. And with that, I'll turn it back to Michelle.
Great, thank you, Dan. So we do have some time for clarification questions for Dan. I just ask you, as I talked about in the beginning, to utilize the questions box and type your questions in. So I will give it a couple minutes for people to type. Connie, I'll give you the slides. For those of you who have asked some questions already and maybe who missed the beginning, the slides will be made available on the Botanical Safety Consortium website as well as the recording. And we'll send an email to everybody who's registered for this, letting them know when those are posted and available. Okay, there is one question for Dan from Jessica Collins. The concern will be regarding patenting supplements. Will this be potential in the future? I don't know if anyone on the group has insight into that. It's not something we've discussed.
I don't think it's anything we've discussed. We certainly, if the question is in reference to the patenting of methods in particular, that would not be the approach of the BSC, because we really want the approach and the toolkit to be available for everyone who is operating in this arena. So ideally, while there may be proprietary methods out there, there would be a common platform in which the botanical safety would be accessible for everyone.
Yeah, and that's something that I think you'll see in the next talk, that this is a public-private partnership and is something that we really do plan to make everything that we work on publicly and openly available. So there's a comment from Bhaskar Mukherjee. Any comments about the history of use in assessing safety of botanicals?
Certainly, I hinted at it in my talk. The history of safe use is a critical component to botanical safety. That history of use, of course, requires a detailed knowledge of the exact exposures that one encounters. So that is where it's important to understand that there's been a good historical record for the type of preparation that's been used for an extended period of time in a focused population or a diverse population. It is a critical part of the safety assessment. When we think about a decision matrix of evaluating a new raw material, it is the starting gate for any evaluation of botanical. Is what do we already know in terms of the history of safe use of that ingredient or ingredients like it. And so when we get into the analytical characterization and the testing, it's always going to be hopefully against a backdrop of an established history of safe use of that material or materials closely like it.
Thanks, Dan. I'm trying to type answers as they come through. Oh, here we go. Sorry, I'm just slowly catching up. Question from Daryl Sullivan. Would a deliverable of the BSc be a way to think about how to conduct safety testing based on the data that this effort delivers?
The goal is to provide a scientific toolkit to provide the opportunity for the characterization of the safety of a botanical. We, as a public-private partnership, we are not expecting that the deliverables will be how that data will be used. That is a separate exercise within a regulatory framework, but our goal would be to create a toolkit that would enable that conversation to occur.
Thank you, Dan. Okay. So one other question related to the recorded webinar, it will be available and the slides will be available on the website, so you could use it. Someone asked if they could use this material as an online college course. We would love for the Botanical Safety Consortium materials to be used more broadly. So, and I will also provide a transcript of the questions and answers, assuming, yes, I will provide a transcript because it will be recorded. I can add those on. Okay, just going through questions here quickly, sorry. Keep up with them all. So there was a point related to, please let the audience know that USP is establishing some public standards for botanicals that are used in dietary supplements, and these would provide safety to consumers. And that's just information from Dr. Srivasan. Kathleen Lee had a question, more specifically, are there labeling rules for different age cohorts for botanicals? Does anyone have any insight there? Anyone want to take that one? I don't know the answer to that. We can follow up on that one. Okay. So one other question, and I think some of this might come up later on as well. Is there sort of a quote unquote Bible of safe daily use levels of these common botanicals in the public domain, a go-to document? I don't think there is, but I don't know if anyone on the group would like to comment on that.
That is often a common struggle that we have when we're looking at botanicals, and it might be an opportunity for some of our international colleagues to jump in here, because I think some of them have had the most recent focused effort on establishing definitions for safe history of use and the evidence for that.
Mm-hmm. That is something. Yeah, go ahead.
Hi, this is Stefan. I tend to point people sometimes to the Botanical Safety Handbook. You won't find all the information there, but I think it's a good start to look at least history of safe use and published data on a big number of important botanicals.
Okay. There is a question from George Parasakov. This is the George. I'm just going to leave it at that. We will talk about the BSC's history in the next talk, so I'm going to hold on that one. And then I think per the question related to age-specific guidelines, you know, I think there was a follow-up question about elderly adults and guidance on dosing, and I think that's something that has come up many times. It doesn't sound like there's anyone on this call who has any insight on specific labeling for age groups.
I think that will be something that we'll address at least superficially in some of the talks following me, but that is clearly going to become a greater interest for some of the technical working groups as we get into systemic toxicology of some of the endpoints and things like that. There are clearly endpoints for which the elderly would be have some increased vulnerability for. So there may indeed be the opportunity to look at whether the assays have the sensitivity for that concern.
Okay. So I will also note in the questions, there are various resources per that other question that people have mentioned. One group mentioned Health Canada and Natural Medicine's database for information. There are some texts available from traditional medicine field. The Botanical Safety Handbook by AHPA is one resource as well. So those are some other ones. And one of the things we're trying to do with the consortium is provide a resource page on our website. It's not up yet, but if you guys, if you have specific suggestions of resources that you're aware of that we could include in that, that would be extremely useful because we do get this question quite a bit. So I had just a couple other questions. This is great. And we might be able to circle back to some of these later. Bernadette Sheridan asks, I see several products with botanicals now being marketed as food. Would we assume the use of a botanical is safe providing in food, I assume, is safe providing it does not exceed the dose listed in the monograph? And I just will jump in to say that within this consortium, we're looking at botanical ingredients. And so the use, and we're focusing on oral exposure. So the use of botanicals in food is in scope of the consortium. And I think the evaluating the dose levels and the different preparations is something that we'll be discussing. Okay. I'm just scrolling through just a couple more. Thanks for your patience here. So Jim McGregor just had a point related to the discussion on the history of safe use, that's emphasized, but there are limitations for delayed toxicities, those dependent on individual sensitivity and secondary factors. So those are things to consider. And I think a lot of what we're trying to do here with the consortium is to help identify additional assays that could help us further elucidate safety. Does anyone else have any other comments on that?
No, other than Jim makes a good point, which is one of the limitations of the data mining for history of safe use is oftentimes one of the hallmarks is the history, the longevity of that use in a diverse enough population to be able to address those kinds of concerns that he's raised.
Okay, great. And then there's a couple more great resources, so appreciate that. Jürgen Schnabel mentions, just as an FYI, that the FEMA expert panel is in the process of reevaluating all of the FEMA GRAS botanicals. So that's another related effort that's happening. Question was, do we consider the ethnomedicine driven processes as well? I think that the potential use of ethnomedicines botanicals is, and if those botanicals would be in the scope and considered by the consortium, I think we're really looking at botanicals more holistically related to oral use. So depending on the particular types of botanicals and the endpoints that we're looking at, I don't see a reason that would not be potentially in scope.
Mhm.
right? Okay, I'm just gonna we're right at time here. Just one more quick question. Any updates regarding facilitating clinical trial assessments of botanicals?
I guess it brings up just a general point of what won't be covered probably today, and that is the BSC has need to have a targeted focus for our efforts, and that focus is on sort of the qualifying with a toolkit of botanicals. However, there are certainly things you've mentioned, you know, this is mostly about oral, but it doesn't mean that the BSC will not have the potential for direct impact upon things like cosmetic products and stuff. And in this case, clinical trials, it's sort of out of scope for the BSC, but we would love for a dialogue to continue in that realm, and whatever we can do to facilitate that would be a satellite kind of effort would be terrific.
Okay, and then one more point from Bodie Timms related to the comment is on with the speed and ease of use, will genetic testing be used with every botanical trial or assay to generate some pharmacogenetic data that might provide a context for some toxicity information? We don't have the plans to do that, but I think discussions related to the value of some of those assays and methods to do that are something we could explore, but it's not currently in scope, I don't think, for any of the working groups. Does anyone have any insight on that?
Certainly something that the Gene Tox Working Group can look at.
Yes, for sure. Okay, so I still have all the questions up. There might be some more that have come in, but I think that we will move on and we can circle back to some of those questions later. So please do not fear if we didn't get to your question. There's a lot coming in. So I'm going to turn it over to my colleague Connie Mitchell, who's a scientific program manager with HESI, who will give us some background on the Botanical Safety Consortium as a public-private partnership and the involvement of our organization, HESI, in convening the BSC. So I'll turn it over to you, Connie.
Great, thanks. Can one of the panelists make sure my slides and audio are okay?
Sounds good.
Great, thanks so much. So hi everyone, thanks for tuning in for the first annual Botanical Safety Consortium meeting. Thanks to Michelle and Dan for setting the stage. I'm Connie Mitchell, and I'm going to talk about the Botanical Safety Consortium as a public-private partnership, and I'll discuss HESI's role. So here's an outline of my talk today. I'll start with some introductions, and then picking up from Dan's talk, I'm going to give an overview of the Botanical Safety Consortium, going through the mission, objectives, and strategy. I'm going to discuss HESI's role in the organization, and then I'll go through the organization structure of the BSC. And I'll end my talk today with a timeline of the year to come. So some introductions. At HESI, the leads on the Botanical Safety Consortium are myself and Michelle. Michelle is the Associate Director of the organization and manages quite a few committees. And I began at HESI in January of 2020, and I primarily work on the Botanical Safety Consortium as a Scientific Program Manager. So trying to pick up from where Dan left off, just giving some history, the former FDA Commissioner Scott Gottlieb made a statement on the agency's new effort to strengthen regulation of dietary supplements by modernizing and reforming FDA's oversight. In his statement from last year, he said, "I'm pleased to announce that we've recently created the Botanical Safety Consortium, a public-private partnership that will gather leading scientific minds to promote scientific advances in evaluating the safety of botanical ingredients and mixtures in dietary supplements." Then later that year, in November of 2019, FDA announced the convening of the consortium as a memorandum of understanding between the FDA, NIEHS National Institute of Environmental Health Sciences, NIEHS, and HESI, naming HESI as the convener of the consortium. And I just want to show a timeline to lay out all the events of the consortium. So between 2018 and 19, the conceptualization of the consortium took place, along with the formation of working groups. And the mission and objectives of the consortium were defined. Then between Q3 and Q4 of 2019, FDA, NIEHS, and HESI discussed and then signed the Memorandum of Understanding, naming HESI as the convener, and a scoping meeting was held late last year to try and define the direction of the consortium. Beginning this year, there was the official launch, the website went live, and we've been adding new members. So with that little bit of history and the background from Dan's talk, the mission of the consortium is to enhance the botanical safety toolkit and bring clarity to botanical dietary ingredient assessment for manufacturers and regulators. And with this mission, we have a series of objectives. One is to engage with a broad group of stakeholders. Another is we will establish appropriate levels of chemical characterization. And we are working to identify in vitro and in silico methods to evaluate botanical safety. We'll compare the tools and the results to the currently available safety information on these botanical ingredients, and we'll integrate what we learn into a framework that will facilitate robust evaluation of botanical ingredients. So those are our overall mission and objectives, but we do have a specific strategy to try and work for the Botanical Safety Consortium. And the first, we will identify botanical ingredient candidates. And this is going to be done by the technical working groups. They're going to select the botanical ingredients based on available safety data specific to those endpoints. And from all the groups, we're going to create a reference botanical ingredient library. Next, using analytical approaches and chemometrics, we will characterize the botanical ingredients. And we will also create a strategy to address some of the other issues, some of what Dan talked about earlier, such as variability between botanical ingredient batches. In concert with this characterization, we will select suitable in silico and in vitro methods that will work for these complex mixtures to evaluate. Once the reference library and assays are selected, we will evaluate the botanical ingredients in the battery of tests. These results will be analyzed and then compared to the existing animal and human safety data. And all these results will be made publicly available. Finally, we will take what we learned about the suitability of these in vitro and in silico techniques for botanical ingredients, and we will see if we can use them for evaluation. And we will identify challenges and prioritize other research, but the ultimate goal is to make a framework for evaluation. So to fulfill this mission objectives and strategy and to keep the consortium on track, HESI was named as the convener. And HESI is the Health and Environmental Science Institute. Our vision is science-based solutions for a sustainable, healthier world. Our mission is to engage with scientists from academia, government, industry, and other scientific organizations to identify and resolve global health and environmental issues. We operate on the principles of creating opportunities to collaborate. We're driven by solutions, we are transparent and independent, and we're committed to excellence. And this graphic shows the different groups coming together, academia, industry, NGOs, and other scientific organizations and government to strive to bring together these sectors for improved safety and innovation for human and environmental health. And HESI has been doing this for over 30 years. And this graphic breaks down our participation, showing our projects are already public-private partnerships. We have over 600 scientists from 250 organizations, with about 2/3 being from the public sector and 1/3 from the private. And the research program funding comes from both the public and private sector. And then this slide just shows some more metrics. It shows some other initiatives, such as memorandums of understanding with government agencies in the US, some global initiatives, and then in the top right is our citation statistics. So through our publications, we've been cited over 20,000 times in over 1,600 organizations across 75 countries. And HESI has many committees, but we have at least 6 that we can directly leverage the expertise for the Botanical Safety Consortium, and they're shown here: cardiac safety, developmental and reproductive toxicology, genetic toxicology, immunosafety, UVCBs, and PBPK models. HESI's many projects are driven through a common mechanism. They consist of a decision framework, data sharing and collective analysis. We sponsor novel experimental studies. We work with other scientists to produce manuscripts, and we work on tools and assay development. And we hold scientific meetings and training sessions such as this one. So what does that mean specifically for the BSc and HESI's role as a convener? We will oversee the scientific program design and strategy, and will manage the different projects, teams, and convening partners. We will leverage expertise from other HESI committees, and will pull data and resources to the groups, and will ensure public benefit and communication through publications and meetings. So with that in mind, I'd like to talk about the resourcing. The consortium is a public-private partnership. There's foundational multi-year funding, which is from the NIEHS and FDA, and there's also private sector funding, which supports additional activities in research. All participants, whether in the public or private sector, will give in-kind support in the form of their time and expertise. But this table shows for the private sector the fees based on annual sales of the company. If a company is already a HESI member, they will not pay the HESI affiliate fee, only the botanical annual fee. However, if they are not a member of HESI already, they will pay both the BSc annual fee and the HESI annual affiliate fee based on the company's annual sales. Trade associations can also join for a flat fee, plus 2 of their member companies must also join the consortium. And these fees apply to members of the Stakeholder Council, the technical working groups, and the steering committee, which I'll talk about more next. I then want to move on to the structure with our role established and some history. So the outer layer of this leaf is the general public, which is open to all. The next layer is the Stakeholder Council. Interested parties must have a justification statement, and companies in the private sector must provide a financial contribution. The next layer in the leaf are the technical working groups. These are the scientific and technical experts for the consortium. And then the last level is the steering committee, which drives the consortium. And I'll go into detail more on all of these. So as mentioned, the general public is open to all for no cost. They have access to publication and website materials, and there is also the opportunity to attend the annual meeting in future years where we hope to have it in person. After that is the Stakeholder Council. This is made up of individuals and organizations that provide input and mission support for the consortium. Members will do this by voting on steering committee composition, nominating scientists to the working groups, and participating in quarterly webinars. Members will provide either direct and/or in-kind contributions, and members can submit proposals and will respond to specific requests from inputs from either the steering committee or the technical working groups. And by definition, all technical working group members and steering committee members are part of the Stakeholder Council. So there's going to be two talks later today on the technical working groups, so I won't go too into detail, but I did want to show this figure that lists our current groups and one we plan to form. So in the center are our toxicological endpoints that we want to evaluate. So we currently have a developmental and reproductive toxicity group, DART, a genotoxicity group, hepatotoxicity with ADME, systemic toxicity with IVE, and we plan later this year to form the cardiotoxicity technical working group. All these groups work in concert with the chemical analysis group to characterize the botanical ingredients the toxicological endpoints will choose. And they'll also work with the data analysis group in both experimental design and in analysis. And for how we pick these topics and how we will consider future topics, there's a couple of factors. We want to think about the severity of the associated disease, such as genotoxicity and cancer, and also based on adverse reporting in the current research. For example, there's a lot of research done on how botanicals affect the liver and hepatotoxicity. We also want topics that will work to build bridges with current animal safety data, So for example, we want to use tools such as an in vitro systemic toxicity model to compare to a repeat dose in vivo model. And with that, all these endpoints need to have alternative assays to measure the effects in the system. So having these in vitro and in silico tools. The last level of organization is the steering committee, and these images show our current steering committee. NIEHS and FDA are the overall consortium chairs, and they're represented by Cynthia Ryder from NTP/NIEHS and by Cara Welch. There are 2 liaisons from the technical working groups, represented by Joe Dever from Amway for systemic toxicity, and by Stefan Gafner from the American Botanical Council, who is the liaison for chemical analysis. Then we have 2 at-large members from the Stakeholder Council, which are Dan Marsman from P&G and Jim Griffiths from CRN. And Michelle Embry and myself from HESI are also in the steering committee. So in case you're wondering where you would fall within the organization group, if you're interested in joining, I have a schematic here that will help to clarify. So an interested party will go to our website and fill out an online application that has basic info and a brief statement on their interest in the consortium. This will be evaluated by the steering committee based on their interest and if they're from the private sector, what they're able to provide. If they don't quite meet the qualifications, they can remain in the general public, or someone who doesn't want to be on the steering committee and just wants to be in the know can also be in the general public. If they do meet the criteria, they can be a member of the Stakeholder Council and participate in the consortium actively. If someone from the Stakeholder Council wants to be on a technical working group, on that same application, they will note their interest and they will give a brief statement of their expertise and submit their CV for the particular technical working group they're interested in. This will be evaluated by the technical working group leads and by the steering committee. And what they'll look at here is the expertise and how they would add to the overall balance of the technical working group. And this has to do with balance in terms of expertise, sector, and geography. If they don't quite meet the criteria, they're still on the Stakeholder Council and are part of the BSC. However, if they do meet the criteria, they can be on a technical working group. So this is our timeline of the year to come. So right now this month, the technical working groups have worked to define their objectives and missions, and additional experts have been contacted, and we're continuing to recruit members. And then throughout May and what we're going to do later on is survey the literature for suitable assays and look for information on botanicals that's already out there for selection. And today's the annual meeting. Throughout the summer in June and July, we're going to collect the scientific literature that's already known, and we'll begin selecting assays and botanical ingredient candidates that we want to nominate. And there's going to be that coordination between the groups that I talked about, between the toxicological endpoints, the chemical analysis, and the data analysis groups. In August, we're going to have an update webinar for our stakeholders. And then in the fall, we hope to finalize the candidate list of the methods and the botanical ingredients. And we want to create literature reviews from what we find on both the ingredients and for the assays. Later this fall, we hope to begin some of the in silico work that would help prioritize the in vitro assays and also do exploratory and method development work as needed. And then next year, we hope to initiate the in vitro assay work with the whole botanical library. I just want to end with our website, which is shown here, www.botanicalsafetyconsortium.org, where you can find more information if you're interested and contact info. And if you would like to be on the Stakeholder Council or the Technical Working Group, you can select Get Involved here, and that will take you to an application form where you can select which one you would like to be on. If you just want general information and be in the public and find out about our publications and upcoming events, on the bottom of the homepage, you can sign up for a general distribution list. And here again is just our website, our general email, and then my email and Michelle's. Thank you all for your attention.
Great, thank you, Connie. So we have some time for clarification questions related to the Botanical Safety Consortium and what Connie just walked through. keeping in mind that we're going to have several additional presentations coming up that'll talk a little bit more about some of the specific objectives and strategy of the consortium. So there is a question related to what the various technical working groups are, and that's coming. Joe Dever is going to be talking about that, but I'll just really briefly mention the ones that we've currently started are looking at developmental and reproductive toxicity, hepatotoxicity and ADME, systemic toxicity, genotoxicity, and then we have a chemical analysis and a data analysis group. And then there's another question, where will the tests be performed?
We are still working to figure that out. It could be something done with our partners, either maybe CROs or some of the industry labs or academics. We haven't quite established that yet.
But we're hoping it'll be a very collaborative effort with the various partners involved. Okay, any other clarifying questions? All right, so I'm going to give me 1 second here. Hold on 1 minute. Okay, sorry for the delay. Our next speaker is Cynthia Ryder, who is at the National Toxicology Program within NIEHS, and will provide some background on the research at NTP on botanical safety. So Cynthia?
Hi, good morning, everyone. So glad that you could join us for our first annual Botanical Safety Consortium meeting. So hopefully you can hear me and see the slides all right. And trying to gain control of the forwarding.
Let me try one more time. You should have, hold on. Okay. Try it now.
Yeah, not quite working yet.
Now, there you go. There we go.
Great. Thank you so much, Michelle. So what I hope to do today is to talk about the National Toxicology Program and our interest in botanical safety testing, and talk about the synergy that can occur between what we're doing at the National Toxicology Program and the Botanical Safety Consortium. So I'll provide a little bit of background on who we are and what we do, and then highlight a couple of projects that I think have a lot of common goals with the Botanical Safety Consortium. So the National Toxicology Program is an interagency program that's headquartered at the National Institute of Environmental Health Sciences in Research Triangle Park, North Carolina. It comes together with the National Institute of Occupational Safety and Health at the CDC and the National Center for Toxicological Research at the FDA. And together we do research on nominated test articles. So we've evaluated thousands of agents in comprehensive toxicology studies. And in addition to our testing, we also have some analysis activities such as the report on carcinogens. And our overall mission is to evaluate agents of public health concern by developing and applying tools of modern toxicology and molecular biology. We do that through a varied toolbox that contains a lot of different things such as data mining, quantitative structure activity relationship profiling. We have bioactivity screening, in vitro studies, short-term in vivo studies in rodents, and chronic in vivo studies. We also use various techniques to do knowledge integration, and all of that is to achieve hypothesis-driven research. We then translate and communicate those findings so that we can provide science to inform decisions that impact human health. This slide is just to highlight that we have recently been trying to be more deliberate about working in strategic areas of focus. And one of those is the combined exposures and mixtures arena. And that is squarely where the botanical work falls. So just important to highlight that we're treating these botanicals as complex mixtures, acknowledging all the complexity and variability that Dan talked about in his slides. In addition to botanicals, we have a diverse portfolio of research, including everything from evaluating cell phone radiation to nanomaterials. So this is part of our larger portfolio of work. Our interest in botanicals at the National Toxicology Program comes from several different motivating factors. First of all, as I mentioned, our mission is to evaluate items of public health concern, and we definitely feel that botanicals are of public health concern because of their widespread exposure, as we discussed, in potentially vulnerable populations, such as people with underlying conditions or the elderly. And unlike many of our environmental contaminant exposures that we're concerned about, these occur at relatively high doses. As Dan mentioned, there can be little safety data on many botanicals, and there's public health concern about the quality and integrity of many botanicals in the marketplace. And in addition to all of that, NTP has received a number of nominations to study botanical dietary supplements. These have come from the National Cancer Institute. There's been some nominations within NIEHS, from private individuals, and from the FDA. So as you can see in this cutout on the right, you can, as a member of the public, submit your favorite nomination to the NTP for consideration for testing. So our interest in botanicals started in the late 1990s. At that time, we really wanted to survey experts and find out where research should be dedicated. So we had a workshop in 1998, and several cross-cutting recommendations came out of that workshop. And I just want to focus in on the first one, which was a recommendation to perform research on potential toxicity associated with high dose or prolonged use of botanicals. So that really motivated the NTP research over the past 20 years. To do that research, we followed the same paradigm we do with all of our test agents. The first step is always to identify the knowledge gaps. So in some cases, it's a specific safety concern like ephedra and cardiotoxicity. And in other cases, it's very general, just a lack of toxicity and carcinogenicity data on a particular ingredient. Next is the test article selection, which for all the reasons Dan mentioned can be very difficult because of the complexity and variability of products. And then our study design in general involves the use of animals, male and female B6C3F1 mice and Sprague Dawley rats typically. And our exposure durations can range from very short term to 2-year. Our dosing paradigm for this group of chemicals is typically oral gavage, but can be dosed feed. And then we have a wide array of endpoints that we evaluate. So clinical chemistry, hematology, we look at genotoxicity, some reproductive endpoints like sperm motility and vaginal cytology, and then comprehensive histopathology. So next, I'm showing a list of completed studies with various botanical ingredients on the left side, and on the right side, some of our ongoing work with different botanical dietary supplements. And here is a chart just kind of summarizing some of the findings from the completed studies. So the top The top ingredients are ones that we did do 2-year bioassays on. And here you can see our calls for the level of evidence for carcinogenicity. So in some cases like ginseng, across the board, there was no evidence for carcinogenicity, milk thistle the same. And then in some cases like aloe vera, under the conditions that these were tested, we see clear evidence of carcinogenicity in male and female rats. So over the course of the past 20 years, we have a lot of experience in evaluating botanical dietary supplements. And through that time, we had a lot of lessons learned. So we really wanted to stop at this point when we had this critical mass of data and look back and talk about lessons learned from all of this work and whether we needed to course correct or continue on the same path. So in 2016, we gathered experts on the NIH campus in Bethesda to talk about addressing challenges in the assessment of botanical dietary supplement safety. And here we wanted to focus on what are the key drivers of research and developing research programs. Where do we need to focus attention? And we really talked about 3 different areas that required research attention. One is comparing across these complex mixtures, botanicals. The second is in identifying active constituents. And the third is in understanding the absorption, distribution, metabolism, and elimination of botanicals. We felt that these 3 areas were really cross-cutting, and they had a great impact on hazard characterization efforts, product development, and regulation. So they'd have a lot of interest from many diverse stakeholders. And this has really driven our research directions since the time of the workshop. So now I want to talk a little bit, just touch upon some of the research areas that we have ongoing now. The first being to develop methods for comparing complex botanicals. So this effort has involved the development of several case studies where we're trying to look across these complex mixtures and better understand how they relate to one another. So here we developed case studies with ginkgo biloba extract, black cohosh extract, and echinacea purpurea. And just to kind of give a snapshot of how we're looking at this here in this semi-busy slide, on the left side, I have an example of a subset of ginkgo biloba extracts that we evaluated using non-targeted chemical analysis. So basically, we purchase a bunch of variations on a theme. So here, ginkgo biloba extract. We do some level of chemical characterization. We also, on those same entities, run them through bioassays. So in this case, I'm just showing one particular endpoint. Here we were looking at a receptor, so constitutive androsten receptor activation through measurement of CYP2B6 gene expression. And you see all of these dose response curves representing the various ginkgos that were tested in this assay. So the critical part is, how do you combine those 2 really different data streams? So here at the bottom is just one method that we've used to try and transparently communicate what we think is similar enough, sufficiently similar, and what we think may be outside of the scope, and maybe the data we're generating on the reference isn't applicable to these different ginkgos, for example. So here on this, just to explain this chart, on the left side, starting at 1, you have the ginkgo that was tested in comprehensive toxicological assays. So that was tested in the 2-year bioassay. And then we converted all of the chemistry data we generated into a distance measure. And so as you move across this chart, you get more and more chemically divergent from our tested ginkgo that has all the accompanying information. So then when we do these in vitro assays, we can also look at how similar the bioactivity profile is to the tested ginkgo. So here we paint that information on top of the chemistry information. So here green means that the bioactivity profile was similar to the tested ginkgo, and red means it was different. Yellow is it was somewhere in between similar and different. And so based on this relationship between chemical profile and bioactivity profile, we can make these clear decisions about what is similar and what is different. So this, I think, is one of the approaches that will be very useful moving forward with the Botanical Safety Consortium, and uses all of this knowledge of complex mixtures and how to evaluate and compare across these. So another area of active research at the National Toxicology Program is identifying the active constituents. So this was discussed at the 2016 meeting as well, and there was a lot of discussion around bioassay guided fractionation. So this is just a visual representation. You start with the raw ingredient, then use various solvents to make extractions. Those extracts can be evaluated in a select bioassay to find the active extract. Next, you undergo separation activities to get different fractions. Again, these can be evaluated in a bioassay to look for the active fraction. The active fraction can then undergo isolation and identification methods to identify either the chemical structure or the class of compounds that might be responsible for the activity that's seen. So here in this example, we're moving forward with a project with black cohosh extract, and this is just taken from one of our papers looking at genotoxicity with black cohosh extract. So currently we're working on applying these bioassay guided fractionation approaches to that compound. Another area where we have active research is advancing these ADME studies of botanicals. So here on the left of the slide, I have listed out some of the standard practices currently. So currently, ADME is rarely assessed in animal studies with botanicals. Often we follow the marker constituent, which may be the marker only based on abundance. There's very little information on drug-botanical interactions. And then finally, animal to human dose comparisons typically rely on administered dose. So our recommendations, which are different from these standard practices, are that we feel that regularly you should assess ADME in animal studies, that we should be following the toxicologically important constituent, so the active, not just a marker constituent, or we should be looking towards newer approaches such as polypharmacokinetics, where you look at the global metabolic profile instead of just one single identified marker constituent. Also, we feel that in silico and in vitro approaches should be leveraged to look for potential drug botanical interactions. And finally, that animal to human dose comparisons should be based on systemic exposure instead of only looking at administered dose. Finally, another area that is ripe for exploration is how do we harness ongoing high throughput screening methods to look at botanicals. So I'm not sure how many of you are familiar with the Toxicology in the 21st Century program, Tox 21. This is a government partnership with FDA, EPA, NIEHS, and the National Center for Advancement of Translational Sciences. Through this program, 10,000 chemicals have been evaluated in high throughput screening assays that measure various biological endpoints in human cells. And so what we've done is used this to pilot a study where we took the entire NTP library of botanicals and put them in a subset of those established cell-based assays in that high throughput screening paradigm. And so this work was published last year by Troy Hubbard et al. And we just wanted to get a sense of the biological activity profiles of botanicals, and if we can put these complex mixtures into this very miniaturized system and get readouts that make sense and that look like we can that this is a feasible approach. So another area that is ripe for evaluation. With that, I just want to say I think there's lots of opportunities to use the lessons learned from the NTP work to inform the Botanical Safety Consortium and to work together towards the common goals that we have. And with that, I'd like to thank many people at NTP that have been involved in the botanical work, particularly Saramia Waidenatha, who's also co-chair of the chemical analysis group, the technical working group. And I'm happy to take any questions.
Thank you, Cynthia. Okay, I'm just gonna pop over to the question box. Okay, give me a second. A bit of an echo. Okay, I'm just going to go through the ones that are most pertinent to Cynthia's talk. All right, so there was a question. Cynthia, might ask you to go back to the slide after the ginkgo leaf, slide 10 maybe. Chi Kim had a question. It's the slide that shows the types of raw materials tested by NTP. And the question is related to how the selection takes place, since there are a diverse range of raw materials available in the market for a single chemical. So how is that determined related to what is developed from what's on the market? Oh, I muted you, Cynthia. Hold on one second. Sorry. There we go.
Okay, that's an excellent question. So we've spent a great deal of time developing an approach for test article selection with complex mixtures, and in particular the botanicals. So I believe Saramia has an entire publication on this for Echinacea purpurea as an example. So generally there are several principles we use to select a test article. One, we want it to be something that people are actually exposed to. So it should be something that is common in the marketplace. It should be representative of what people are being exposed to. So if there is a NIST standard reference material, we prefer for what is tested to look like that NIST standard reference material. And then, yeah, so it's been recommended in the past that perhaps we should extract our own material in order to have it, to know exactly the process by which it's made. But the problem with that is then we'd be introducing yet another variable, and people might not be exposed at all to something that we did. But it differs case by case, but generally we do chemical analysis on multiple lots and variations, and then we compare to some high quality standard reference and select based on that. So since we've started doing the sufficient similarity work and comparing chemistry and biological activity, we've introduced that into our test article selection process, so that we're not only going to be basing our selection on similarity of chemistry and being representative of the chemistry, we also want to introduce that biological component in the selection process. That was a very long answer. Sorry about that.
Thank you, Cynthia. So sorry, I'm just going through here. So one question was related to, in Dan's presentation, Dan talked about the fact that animal testing in many cases is poorly suited, but the animal testing was largely the subject of NTP's work and your presentation. So could you comment on that going forward? And then whether the BSc will be focusing on animal or non-animal approaches.
Yes, so I think animal work continues to be the gold standard for the safety evaluation at this moment. We have not gotten to a place where we have replaced animal testing with a more predictive of human health approach. But what we're trying to do is build the case for using alternative in vitro in silico approaches that can be helpful in reading out safety. So the Botanical Safety Consortium is focused on the in vitro in silico side completely. It will not be doing animal testing. The National Toxicology Program, on the other hand, continues to use animal testing as part of our testing portfolio. And so some questions with chronic toxicity may require additional testing by other entities, but the Botanical Safety Consortium is focused completely on building the toolbox of in vitro in silico approaches.
Great. Sorry, I'm just typing. One other question, and I'm gonna put this one towards you, Cynthia, that's come up probably with 3 different questions, was related to the definition of safety for the purposes of the Botanical Safety Consortium. And I don't know if you want to address that, since you were sort of one of the first initiators of the consortium, and I think there's a lot wrapped up in that question, of course.
Yeah, and one other thing I want to say about the animal to in vitro comparison is, I think I talked about our animal work at NTP to highlight that we have this store of animal data to compare to the in vitro work that comes out of the Botanical Safety Consortium. And so I think there we're trying to build those connections and strengthen confidence in the newer approaches by having that data to rely on, and also looking towards human data such as the adverse event reporting and clinical trials that have available toxicity data. So about safety, how do we define safety? I think it's, I mean, that's a tough question, but I think it's doing our best to evaluate these as thoroughly as possible. And I think it's important to look at where we are now versus where we want to be. So if we rely completely on history of safe use with no other evaluations, that's kind of where a lot of things stand at the moment. So we're wanting to go from that place of very little testing to increased testing using these newer approaches, so to increase our safety data that's available. I think it's impossible to say that we can comprehensively guarantee safety for anything, especially considering the complexity and variability of these substances.
Thanks, Cynthia. And I think this gets at that point as well, related to ensuring that the testing that we undertake within, and this has come up in several of the working groups thus far, that we're evaluating not just the hazard, but of course looking at the doses that are commonly used for the products on the market, so that we're able to look at both parts of that equation. So I think we're looking at assays that are going to help us identify the toxicities, but keeping the relevant doses and the types of preparations that are on the market is in scope of the consortium as well. Anyone else on the steering team have comments related to that? Okay, because we are also committed to looking at the relevance of, again, what people are being exposed to. So there's been a couple questions related to the ADME. I'm gonna just, there's some long questions, so I'm just gonna kind of pull this up. I guess, sorry, hold on one second. So Jan Q had a question, curious about the recommendation on ADME in animal studies. As we are advancing in alternative testing, is this still a timely direction? Would alternative methods for ADME evaluation be an area of emphasis for the Botanical Safety Consortium?
Yes, most certainly. And I think the systemic toxicology group is looking at things like IV, IVE. So definitely, I would say my comments on ADME and animal studies were about comparing across doses, extrapolating from one route to another, using the existing body of safety data on animal studies to inform and translate to human safety. But yes, we are definitely exploring the in vitro capabilities and looking at how to extrapolate from in vitro to in vivo.
And I'll just add on to that, we've had discussions within the hepatotox group as well and others and the systemic about forming a specific sort of smaller task team that's particularly focused on ADME. And this gets to another question related to that that Bhaskar Mukherjee asked, related to the strategies to overcome that challenge in identifying and evaluating the dose delivered when you're looking at testing mixtures and extracts. And Saramia's work has been looking at this, and I think that's a key part of the BSc that we'll be working on, potentially in a smaller separate subteam specifically focused on ADME. And I think that also gets at a question that was asked about polypharmacokinetics. How is the analyte defined? How is that relevant if it's a mix of ingredients with differing functions and benefits?
Yeah, so I think we're just beginning to explore polypharmacokinetics as an option. So we still have a lot to learn in that area, and I don't want to step out too far on the limb to talk about the details of what exactly it will say. I will say in our early phase, we do have a project where we're trying to compare the polypharmacokinetic results to the traditional ADME approach and see what we get, how it compares, and look at whether it's a viable approach for moving forward with other botanicals in our program.
Great, thanks, Cynthia. Another question, and maybe this is, I don't know if this is something you've explored in NTP, related to work on similarities and whether there have been any algorithms or technology developed for predictive purposes related to, say, compounds of significant similarity.
So if that's talking about the statistical comparison of the data, or is that talking about predicting based on structures that are known? So I could say about the statistical approaches, yes, there are several methods that are being employed by different groups. Interestingly, there's other projects in areas such as drinking water disinfection byproducts or pesticides present in childcare centers. You know, there's other groups that are working on this question of sufficient similarity and how you, I think the important part is how you justify that decision that something is sufficiently similar or is different. And so I think we are very much following the literature and new developments in that area. It's an area of great interest for us and for others. So yes.
Great. And then there's one more question. I think we've been talking about this a little bit from Eric Secor. Will there be an effort to assess current raw and finished materials already in the marketplace? And related to that, would the BSc be partnering with stakeholders to help assess the current rate and quality of testing and labeling and things like that? I think the first question maybe is more critical here, related to currently what's on the market and different preparations available.
Yeah, so I think that's a very important part of any evaluation of sufficient similarity, is to get a diversity of materials that are the same ingredient. So in all of our case studies with ginkgo and black cohosh and echinacea, we did both the raw material as well as finished products that we just ordered, you know, from the marketplace. So as well as any available standard reference materials. So yes, I think that's a key part of looking at whether, for example, we have this conversation about, do the excipients contribute to any observed toxicity, teasing apart what is driving the bioactivity. So I would say that is important.
Okay. And then I have one more NTP specific question. How, and maybe you covered this, but how does the NTP prioritize those ingredients?
For testing, yes.
Yeah.
So once they're nominated, we look at the widespread exposure. So our first and foremost is we don't want to invest your taxpayer dollars in a very obscure substance. We definitely want to test something that has the most widespread exposure possible. And then we look for any signs that this is of public health concern. So if there are adverse events reported around a certain ingredient, that will increase the priority for testing. So if we get a specific ask from a regulatory agency, that increases priority for testing. So we make decisions on a multifactorial basis.
Okay. Okay, and then there were a couple questions related to non-oral, which is outside at least the current scope of the BSc. But there was a question of whether or not, sorry, I lost the question here. NTP has approaches that have been used to look at absorption, at dermal absorption for botanicals.
I'm trying to think. I don't think we have done anything in NTP. I'm not sure if NCTR, our partner at FDA, has done some of that work, and I know NIEHS often does dermal work, but I'm trying to think specifically to botanicals. I can't think of anything off the top of my head.
Okay. And then one other question related to some of the studies that have occurred at NTP. Have those been related, and this is again getting at the what's on the marketplace, current dose levels, the characterization of substances on the marketplace. So for the studies that have been done, the in vivo studies at NTP, is there an effort to then compare the data from those back to not only what's currently in the market related to doses, but also the chemical composition?
Okay, I just got a message from my boss that aloe vera phototoxicity was dermal. So that's a study on aloe vera that was done at NCTR. Thank you, Nigel. And so I kind of spaced out on the question at hand.
I can ask it again. No, that's fine. I was wondering for the studies that have been completed at NTP, where you have data, has there been an effort to compare the extracts or whatever the botanical ingredient that was tested in those studies to what's currently on the marketplace with relation to the dose as well as the chemical composition of what's sort of currently used by the general public?
Yes, that was another reason that for the ADME recommendations, for example. So part of the work on the ginkgo example, we did compare a ADME study or a TK study with a specific known in the marketplace ginkgo EGB 761 to our ginkgo to look at whether the profile of the toxicokinetic profile was similar. So yes, and then also, for example, with the aloe vera project, a specific constituent was tested alone to confirm that the toxicity observed was due to that constituent. We saw the same, and this was more work done at NCTR with aloin. And so when you do that kind of work, then you can, and link to a specific constituent, then by monitoring that specific constituent, you can compare very easily across variations with that constituent. So in certain cases, yes, and often we, in our in vitro studies, we test single constituents that are the purported actives alongside the complex mixtures to better understand what the drivers of toxicity are. So but that is an important part.
Okay, great. And I'm just going to do one more question before we turn it over to Cara, because this has come up a couple times. And maybe this is something we can circle back to at the end and and Cynthia, you could speak to whether NTP's done it, but any of the other panelists could also answer this question. What work do we foresee related to botanical interactions? Because a lot of these products that are on the market don't contain one botanical, they contain multiple botanical ingredients. And so how that would be evaluated.
That's a great question. So, you know, complex mixtures aren't enough. Let's do complex mixtures on top of complex mixtures. Yes, but that is so important. And I think a lot of the work on, you know, those complex ingredients like in Hydroxycut or the Oxy Elite Pro that are being worked on, things like energy drinks, these are all important questions. And I think the same principles of mixtures toxicology with one complex mixture, you can look at those principles with multi-ingredient complex mixtures. So you can do regression analysis where you vary the ratios to understand which of the ingredients is driving any observed bioactivity. So I think, yes, we need to put our mixtures caps on and think about these things and not stop at single ingredients, because the real world, we're exposed to these multi-component mixtures as well as, you know, the surrounding environment, which contains a billion other things. So definitely.
Okay. So with that, I'm going to turn it over to Cara Welch to talk about the FDA's work. So I can see your slides, Cara. It looks like you're all set.
All right, thank you, Michelle. And thank you all. I'm very pleased to be here with everyone today, virtually, of course. In line with Connie and Cynthia's talks, I'm here to talk about FDA's role with the Botanical Safety Consortium. And I am pleased to look at it through the lens of our more recently announced plan to modernize dietary supplement regulation. So, you know, brief introduction into FDA's dietary supplement program, moving into our modernization plan, and then how we see the consortium fitting in. So thank you very much. Our Office of Dietary Supplement Programs directs FDA's activities specifically regarding dietary supplements. We provide the policy perspective for dietary supplement activities. You know, ideally, all policy documents that affect our program come through our office once, twice, or three times. We, of course, are involved in reviewing safety, ingredient safety, and dietary supplement safety. We process current good manufacturing practice compliance actions, that's CGMP, misbranding, so the labeling of products, and then disease claim compliance actions. That's actually a big part of our office activities. We're fairly active regarding dietary supplement compliance. And then we also provide expertise for civil and criminal cases as needed. And we have these activities, we undertake these activities working through our priorities. So we established these ODSP priorities a number of years ago, and they really help us sort of focus our day-to-day activities, but also our longer-term activities. First and foremost, our priority is to protect consumers from unsafe products. And that priority, of course, directs a lot of activities throughout the agency. It also reprioritizes a lot of activities should something come up. We also spend a lot of time on ensuring product integrity. What we mean by product integrity, we're getting at quality of a product, a lot of the CGMP and misbranding aspects regarding dietary supplements. So a product that is labeled as a dietary supplement, is it labeled as, ensuring product integrity, a product that is labeled to have certain ingredients? Does it have those ingredients at the level it is supposed to have those ingredients and nothing else? That is what the product should be. And then we also work on promoting informed decision making, which is our way of saying we want to ensure that people who are choosing to take dietary supplements or recommend them to their patients, do consumers and healthcare providers understand what it is that they're taking? So what is a dietary supplement? Should a dietary supplement be? What should it not be, of course? It should not be claiming to treat, cure, or prevent a disease. It should not, there are some limitations in the United States as to what a dietary supplement is and is not. It is not an inhaled product or an injected or topical product. And so as they go out and choose what product they want to take, making sure they have the information they need at hand. So our program has been in place, of course, for a number of years, at least for the last 26 years since the passing of the Dietary Supplement Health and Education Act. But last February, we announced our plan to sort of modernize dietary supplement regulation. We announced this through an FDA commissioner statement. Connie mentioned this earlier. And our statement really focused on or highlighted 5 specific areas. And I'm actually going to use these 5 sections as I go through the rest of my talk. So, we highlighted the need for enhanced communication with our stakeholders, consumers, industry stakeholders, federal partners, of course. We announced a commitment, or rather a recommitment to enforcement. I already mentioned that's an area we're already pretty active in, but are we approaching it in the right way? We announced we want to take a flexible approach to our regulatory framework. We need to be sure that our framework fits today's dietary supplement industry, but we all know the industry moves very quickly. So is it flexible enough to address tomorrow's dietary supplement industry as well? We used the statement as an opportunity to kick off our engagement in a public dialogue, specifically around potential legislative tweaks to DSHEA. And then specifically with the consortium, we announced our commitment to partnering with our stakeholders to protect public health. So first, talking about enhanced communication, and more so a new approach to how we're communicating about ingredients that aren't being lawfully used in dietary supplements. This is a quote from the commissioner's statement in February 2019, announcing a rollout of a new ingredient advisory program. So the ingredient advisory list, we officially rolled this out in April 2019, and it announced the creation of a rapid response tool we're calling the Dietary Supplement Ingredient Advisory List. This is a list that lives on our webpage. It gives us a way to communicate more quickly and more efficiently when we have concerns about an ingredient that is being used in dietary supplements. Specifically, these ingredients are ingredients that FDA has looked at and preliminarily determined they do not appear to be lawfully used in dietary supplements. It could be for a number of reasons. It could be the ingredient doesn't actually fit the definition of what is a dietary ingredient, but it's being used as a dietary ingredient. It could be that it's a new dietary ingredient, or NDI, for which a pre-market notification is required and hasn't been submitted. The use of that ingredient in a dietary supplement would render the product adulterated. So we periodically add ingredients to the list, and then as we finalize determinations, we will also be periodically removing ingredients from the list, because this is only for ingredients that have preliminary determinations. So if we finalize the determination that it is in fact not a dietary ingredient, it shouldn't be on this list anymore. It should move somewhere else on on our website, ideally. To be clear, inclusion on the ingredient advisory list is not a statement about the ingredient safety. It's purely on the regulatory status of the ingredient in dietary supplements. But it is a great example of modernizing our approach. The industry moves quickly, novel ingredients are introduced to the marketplace, and these novel ingredients can raise complex questions involving science and policy and the law. And in the time that it takes us to make a final determination about the status of these ingredients, consumers and industry might conclude that a lack of public action by FDA indicates the ingredient is being marketed lawfully, when in fact it is not. So we want to have the opportunity to communicate our preliminary assessment. And then based on this information, dietary supplement firms may choose to avoid using this product or this ingredient in their products. Consumers may decide to avoid using the products. Also, firms may decide to provide us their information as to why they believe it is being lawfully used. And so I think that's a great opportunity too. We review the information we have at hand. We may not have all the information, so it's always helpful if firms can provide us their information. Talking about enforcement, again, sort of a recommitment to enforcement and ensuring that our approaches are appropriate for today's market. We want to develop new strategies and new approaches to enforcement. And the enforcement, I think it goes, activity goes beyond just traditional enforcement activity. We do, of course, have a number of claims-specific compliance actions. Compliance actions in FDA speak generally rely to warning letters. We will periodically send out a batch of warning letters on a particular claim. So when we announced our plan for dietary supplement modernization in February of 2019, we rolled it out with a number of warning letters for products making Alzheimer's disease claims, which is it's really not appropriate. We will periodically do claims initiatives for cancer or, other really serious diseases that just should not be part of the dietary supplement industry. And I know everyone agrees with this. Of course, for the last couple of months, we've been focusing our claims-specific compliance actions on COVID-19 claims. So we and FTC have been fairly active on that end. We will also periodically send out warning letters on specific ingredients. So once we have a final determination, we can send out a warning letter specifically warning a firm that their product, is found to contain this ingredient and that is inappropriate. So in April 2019, we announced a number of warning letters regarding DMHA. And that is, I think, a great example of why our ingredient advisory list is, an approach that's really needed. DMHA had been around for a while. It took us a while to finalize our determination, but we don't want to imply that it was appropriate for however long it was being used. We also sent out ingredient warning letters on the use of phenibut in dietary supplements, which does not fit the definition of a dietary ingredient. So it shouldn't be used as a dietary ingredient. Judicial actions are usually going to be injunctions. We will often have injunctions for CGMP or current good manufacturing practices. That's sort of getting at the manufacturing practices of the product, as well as claims. At the beginning of the year, beginning of late 2019, early 2020, we announced an injunction, a consent decree with ABH, Nature's Products, that had a far wide-reaching effect throughout the industry. It was a contract manufacturer. We also last year, I think it was last June, announced an injunction of a seizure of a firm's product, Life Rising, that was due to, again, manufacturing practices, but also toxic element levels. So, there are times when it has a very direct safety link, these GMP concerns. But we will also take activity outside of sort of the traditional enforcement activity. We've issued a few safety alerts in the recent year or so. In February of 2019, we issued a broad safety alert for all consumers to avoid dietary supplements containing cesium salt, specifically cesium chloride. Thankfully, I don't believe it's a widely used ingredient, but still it was important that we get the information out there. And then last summer, we issued a safety alert for the use of vinpocetine in dietary supplements. That alert was more focused on women of childbearing age, since that was the population most at risk that we're aware of with the use of vinpocetine. So talking about a flexible regulatory framework, this is sort of getting at the idea of ensuring our program is adaptable for today's dietary supplement market, as well as tomorrow's and next year's, while still promoting innovation. So we started off this discussion with a public meeting, an FDA public meeting last May, about a year ago, which was, I think, actually entitled Responsible Innovation in Dietary Supplements. And so we really wanted to dig into how FDA can better allow for innovation and growth in the dietary supplement marketplace, while maintaining and even strengthening our ability to evaluate product safety. We discussed topics largely around NDI, or new dietary ingredients, but what kinds of ingredients can be used in dietary supplements, whether and how the growth of the marketplace has impacted the existing framework, including examples specifically to when a notification, a pre-market NDI notification, is not needed, and then how to promote overall compliance with the pre-market notification requirement. So we sort of dug into, are there incentives that we can provide? Do we need greater enforcement? Or, as some of our panelists discussed, a combination of the two. And then, sometimes we have issues that deserve a broader discussion, like the public meeting. Sometimes we have discussions that play out on the smaller scale. So we continue to have stakeholder discussions on topics such as master files, including a recently submitted proposal from some of the dietary supplement trade associations specifically on this topic. We also continue to have stakeholder interest in the development of an ODI list, or Old Dietary Ingredient List. This was the topic of a public meeting a few years ago, and as needed, we continue to discuss the merits of such a list and whether it's important that FDA develop such a list. And then engaging in a public dialogue. In particular, we wanted to, so we used our February 2019 statement to engage in a public dialogue of whether changes to DSHEA would strengthen the industry while still preserving the essential balance that DSHEA established, allowing consumer access to safe, high-quality products while upholding FDA's ability to protect the public from unsafe and otherwise unlawful products. There's a balance there. We recognize that, but we do think there are changes that can occur to keep that balance. So we are aware, of course, that certain parts of the dietary supplement industry have been discussing changes to DSHEA for several years. FDA formally joined the discussion in March of last year when FDA's budget request went public. It included a proposal for mandatory product listing for dietary supplements. This is specifically a proposal to require all products marketed as dietary supplements to be listed with FDA and to give FDA authority to act against non-compliant products and the manufacturers and distributors. So there are, of course, any number of details that go along with this type of proposal. and the details are not yet set in stone. We do want to have a discussion, a negotiation if need be. It's important from our perspective to engage in the discussion because we really do believe this type of move would put us in the right direction. Product listing program, collecting even the most basic label information, will enable FDA to identify and act against illegitimate, dangerous, and otherwise illegal new products more quickly by the simple fact of getting us past the barrier of knowing these products exist. We believe it will improve transparency and level the playing field for a responsible industry by putting all the information out there for everyone to see. Consumers, retailers, and of course FDA can then easily see whether a product has taken the most basic steps towards compliance. Are they opting into the industry by listing their product? Additionally, FDA, by having this information at hand, will be able to better utilize our limited resources through a more strategic and risk-based approach for things like research or facility inspections. Are we inspecting the right facilities? And then this past spring, March of 2020, FDA included another legislative proposal in the budget request to close what we see as a loophole in the drug exclusion provision. It gets a bit technical, so I apologize, but there are certain ingredients that have been authorized for investigation as a new drug and for which clinical trials have been instituted and made public. That's written in the statute if you need the details. Or there are ingredients that are approved as a new drug. These ingredients are often excluded from the definition of a dietary supplement. That's part of the definition of what is a dietary supplement. It does not include certain ingredients. However, we've found there are, likely a very limited subset, but a limited subset of ingredients that were once, but are not currently approved as a new drug, but also preceded the IND requirement. And it seems these ingredients fall into a loophole that we don't believe was intended by the DSHEA drafters. So our goal here is to make a simple fix to close that loophole and better protect the integrity of what is a dietary supplement, or more appropriately, what is not a dietary supplement. So these two proposals are intended to improve upon the base that DSHEA provides. And of course, we're looking forward to continuing dialogue on these issues. And then the consortium, the BSC, partnering to protect public health. Announcing the consortium was a big part of the February 2019 announcement. Protecting public health is something that's so important to each of the parties involved. You've already heard it from several presenters. You're going to hear it for the rest of the day. But I think everyone agrees that we can be more efficient and effective working together. And I'm really so happy to be part of this group. So the consortium was established. Connie already gave you sort of the brief history. I was not actually intimately involved in the development of the BSC. I was lucky enough to know what was happening, was confident that it was in good hands, and I didn't need to get into the details. And let me tell you, there were a lot of details to work through. Collaborating with the federal government is not an easy task. So kudos to all my steering committee members for pushing this forward. And I'm even luckier to join the group now that the heavy lifting is complete, or I should say largely complete. The work continues, of course. So yes, as Connie explained, the consortium was formally convened in November of 2019. The executed MOU between FDA, NIEHS, and HESI. So this group has brought together scientific experts from government, industry, academia, and others to advance the science of botanical safety evaluation. The data that's generated by this effort is really important to FDA's regulation of dietary supplements. To be clear, it does not provide a direct link to regulatory decisions. It's not intended to suggest the toolkit developed by the BSC must be used to get a passing grade from FDA. However, from our perspective, we hope it will allow FDA to leverage our existing resources and our own evaluation of products and ingredients. We want to benefit from the use of this toolkit. And of course, the data generated will provide a benefit to dietary supplement firms as they are operating in this industry. So as they go through the process of evaluating the safety of new ingredients and products that they're looking to market, this toolkit is valuable. So I think from our perspective, we see this as a rising tide lifts all boats type of effort. So with that, I am done. Michelle, I turn it back over to you.
Great. Thank you so much, Cara. So I know we're running a little behind schedule, but there's some specific questions for Cara, and I checked with my other speakers and they were okay with us taking some clarifying FDA specific questions. So we're going to go through a few of those before we move on to our next speaker. So there's one from Jim McGregor and several on the same topic. Has the FDA finalized its guidance on safety information expected through the NDI for those marketed prior to 94?
Sure. So it is still a revised draft guidance. The last revised draft guidance was published in August of 2016. That is the most current document. And
that one is publicly available.
It is publicly, absolutely, yeah.
Thank you.
And just to add on to my answer, it is important for us as we are continuing to move forward. It's a behemoth document, so I think we are trying to move forward strategically to provide as much clarification as we can. As always on new dietary ingredient questions, and there are always a lot of new dietary ingredient questions, we really do want to be a resource. The revised draft guidance from August 2016 is a great resource. I have it regularly. I pull it up very regularly. I have it bookmarked. But also, if there are questions that you're just not seeing, if you have questions that you're not seeing an obvious answer to in the guidance, that happens. The guidance document is not written for everyone's specific situation. It's sort of written for the mass. I would absolutely encourage you to come to FDA and ask the questions directly. You can contact me. My email address is just my name. It's cara.welch. You can email the email address on the screen right now, and we will get it to the right SMEs to make sure you can get an answer. And then we do encourage firms, if they are interested in or working towards submitting a new dietary ingredient notification, if you have questions, our team is available for pre-NDI meetings before you actually submit the notification. If you have questions, I get a lot of great feedback from those meetings. So I think there are resources available, but right, the revised draft guidance is the current document.
Okay, great, thank you. There was another question, I think you covered this on your last slide, but it was related to, is FDA considering mandatory product testing for registration?
Product testing?
It says, is FDA considering mandatory product, sorry, I see, I read it wrong. As FDA is considering mandatory product listing, is product registration also a point for consideration?
Well, I think that depends somewhat on different terminologies, and I think that's just a great point to sort of dig into is, people have a lot of, with certain terms that we use, sometimes there are impressions that come along with it. So, we use the term product listing. We are talking about listing your product and sort of basic information about your product with FDA. It is not an approval process. We fully agree that DSHEA did not intend to FDA to have an approval authority. It's not something we're asking for. But it's also not going to be the same as, it would be a separate system than facility registration. And so to an extent, we're using a different term to sort of distinguish ourselves from the food facility registration requirement, which applies to some people who would list their products, but not necessarily everyone. Some people would be required to register their facilities and wouldn't necessarily have a product that they need to. And so there's some sort of distinction. It's like a Venn diagram, right? There's some distinct categories and some overlap. But I think engaging in the dialogue is why this is also important, because what we don't want is for people to be afraid of what FDA is asking for when it might just be we're using the wrong terminology. So I think we're really excited to sort of dig into the details with our stakeholders and say, this is what we think is important. What do you think is important? And where can we meet in the middle?
Thanks, Cara. Two more quick questions, maybe not quick answers, but Rupesh Gupta mentioned the point that when in Dan's presentation and also in Cynthia's, that there's considerable, when there's products that have high sale in the market and NTP prioritizes chemicals for testing, but there's gaps for a robust in vivo safety, even if there may be history of safe use. And does that impact FDA's vision and policy thinking on in-market current commercialization?
Yeah, so I am not totally sure I have a good answer. And I'm not actually sure I understand the question. So I'm not even sure it's worth me postulating for a while. But I think this is something, if you wanted to send an email, I bet I could answer it, or at least get you in the right direction. I'm sorry.
No, that's no problem. I think it's sometimes hard to write all these questions. So Rupesh, please clarify your question or send an email and we can try to get the answer to you. I think what the point is related to known gaps in vivo safety testing and how that plays into kind of the FDA's vision on botanicals on the market, just more broadly.
Got it, yes.
Okay. Sorry, I'm just trying to see. One question was, would a master file approach be considered for the ingredients in order to facilitate commercialization? And I'm not familiar with that term. Maybe that's something used at FDA, Cara?
Yeah, I think this is another one of those terms that can mean something a little differently for various stakeholders. But a master file system is very broadly, there's always specifics that go along with it, but generally it's in the drug sense, it's used as sort of a repository of information on one aspect of a total drug product. So the master file for this excipient is sort of on file with FDA, and as people use that excipient, they then reference that master file because it's kept on file with FDA. And I apologize for giving the example in the drug sense, but we don't have a master file system established for dietary supplements. We are open to the concept, and I think I already referenced the fact that we've received a more formal proposal from some of the trade associations. Still reviewing that. No specific thoughts on that yet, but we have certainly given it a lot of thought over the last couple of years. We've had a lot of in discussions with the various stakeholders on the topic, and I think we still have, some people use the term differently than other people, and so I think what we want to do is be very clear, if we do establish a system, what would benefit our industry and still be within FDA's abilities, within our sort of regulatory and statutory structure. What can we provide? I don't know that a master file system, I think the work of the BSC and the toolkit that they can provide, will be providing, will certainly be helpful in sort of what we would like to see is that it will be helpful in sort of putting together a safety package for the use of a particular ingredient in a particular product. I think that were a master file system to be established, again, I would hope that would be one of the ways in which it could be utilized, but by no means required, because I think FDA does want to be very clear that the work of the BSC is something we feel very strongly in, but there won't be sort of that direct regulatory link.
Thank you, Karen. I think that gets to the last question, which is more specifically related to FDA's role in the Botanical Safety Consortium, related to how it will use or intends to use the collaboration related to safety reviews and how the data generated and the toolbox that's developed from the Botanical Safety Consortium might be used to help inform FDA's reviews or decisions. So I don't know if you just want to speak to that a little bit before we move on.
Yeah, absolutely. So we definitely are looking forward to using the toolkit as much as the rest of our stakeholders. So I think what we don't see it as is a requirement for FDA reviews, but what we do hope to see is FDA can benefit from it as we do our evaluations. We want to see that link between the chemical characterization linked to assays, in vivo, in vitro assays that are out there. FDA has also publicly talked about our desire to, reduce unnecessary testing, animal testing. We're working on that as well in sort of the broader sense. So I see that fitting in, the work of the BSC sort of fitting into that as well. And I think the part I'm most interested in is just another important tool for industry as they're going through the process of evaluating ingredients. I can only imagine it's daunting, to say the least, to look at these very complex extracts and how to be comfortable that you are putting out a product that is reasonably expected to be safe. Complex on top of complex, as Cynthia described it. So I think that is something I'm really hopeful we can have in place for everyone's use.
Great, thank you, Cara. Okay, I think we're going to move on. I know there were a few other questions and we'll try to circle back to those at the end, so don't fear, we haven't forgotten your questions. So our next speaker is Joe Dever, who's at Amway, and he is going to get us into some more of the details related to our technical working groups. So over to you, Joe.
Thank you, Michelle, and it's a real pleasure to get to really introduce, I think, what is really going to be the driving force of the consortium. It's these technical working groups. This is where the nuts and bolts of the work, the detail that's needed, this is where it's all going to get done. So I'm really excited to give this talk. And first, I just want to thank Connie Mitchell. She put a lot of these slides together. I want to make sure she gets credit for that. So let's jump right into it. I really just kind of want to give everybody an overview of these groups and what it's like to be a part of them. So as I mentioned, these are really the core technical entities responsible for designing, executing, delivering on BSC's scientific mission. So again, focused on specific scientific questions related to the evaluation of botanical ingredients. This is obviously, this is where the great amount of work is going to get done, and it's going to take a lot of different types of expertise to get there. And as you're going to see, we're very, very fortunate to have already acquired a lot of really outstanding experts and a diverse group of people trying to tackle the very difficult issues that come about with botanicals, as many of the speakers have already indicated. So what does being a part of a technical working group involve? There's teleconferences, right? This is where strategies are discussed, data is discussed, and ideas come forward. Providing technical and strategic input and perspectives during the working group's ongoing scientific discussions. This is the place for research. This is a place for designing experiments, data analysis. This is the place for writing and also communicating in specific technical areas on behalf of the working group. And really, in addition to the core working groups themselves, there will likely, and there already are, going to be subteams that freak out in specific areas. And there's just so many different aspects of work that goes into botanicals, and that's what makes them challenging. It's difficult, and so these little subgroups are going to break out to try to tackle specific areas within a specific endpoint. So just a word on how technical working groups have been put together and will be put together in the future. So there's really a number of different ways this can happen. I mean, they can be nominated by the steering committee, by stakeholder council members, technical working group members themselves, or staff. And eligibility is really an evaluation process based on a lot of different factors. Certainly, scientific and technical expertise within the particular working group is probably the most important criteria. I think that, you know, as these working groups are beginning to solidify their strategies and the types of things that they need to hit, there's going to be specific gaps in areas that are identified where experts will be sought at to fill. So one of the core principles around the BSc, but also around the working groups, is to strive for balance, right? We really want to have a great balance of academic, government, public, private, all these different areas. We need experts from all of these areas to come together to solve these very challenging technical problems. Geography as well is really important. We want to have access to experts around the world as well. And again, membership is evaluated on a yearly basis, and we expect that there'll be some ebbing and flowing as people come in or people opt out, and that's perfectly fine. It's a dynamic process. So this is a slide that you've already seen a few times, so I'll go through it quickly in case you missed it. But this is just really the core objectives of the consortium: engaging with a broad group of stakeholders. Chemical characterization is a really important aspect. And again, identifying those fit for purpose in vitro and in silico assays to evaluate botanical safety in a pragmatic manner, and applying these tools in the context of currently available information. And again, that gets at this idea of history of use and how that connects to the development of these tools, integrating them into the framework. More on the strategy. Again, I'll go through this pretty quickly because I think it's been covered well by some of the other folks. Identifying candidates, right? Identifying botanicals. And again, the working groups are going to be critical in this process. And different botanicals may have different types of toxicities that may be present from them, or may be good candidates to look at certain specific areas. And then different working groups may select botanicals focused on their working group. There'll be an effort at characterization, and then suitable approaches and assays will be identified, evaluation of a library, analysis and reporting, and then this framework and toolbox development. And again, I think that this has been covered pretty well, so I'll go on pretty quickly. The working groups are obviously intimately involved in every aspect of this. This is another slide you've seen as well, but I love it, so I'm going to show it again, because I really think it gets at how the working groups, not only the areas that they're covering, but how they're working together and how they're integrated as a whole. So on the outside, you see that chemical analysis and data analysis obviously are a part of all the working groups. And they're really going to be, in a sense, support groups that help the other working groups, genotoxicity, hepatotoxicity, systemic toxicity, analyze their data, get chemicals that are well characterized so they can do their work with confidence. So this is all linked, and I think that's one of the most important aspects of the consortium, is that we're not going to be siloed. Obviously, each working group is going to work in its area, but they're going to be connected to other working groups. They're going to be communicating with other working groups because there's overlap. And I think that's what this slide represents. And I think it's that sort of cross working group communication where some of the best work is actually going to get done in the consortium. I wanted to take a moment, again, just to sort of give everybody an idea on what we're trying to do in terms of the paradigm. And this has been hinted at by Dan and others in terms of how we integrate history of use into a testing paradigm that might help fill gaps. So this is a hypothetical graph of 5 botanicals with varying levels of history of use data. Botanical number 1 has enough history of use data to substantiate it. And that's a very common occurrence, and many botanicals do have that level of history of use data. But there are others that may have certain gaps, or may have less and less varying degrees of history of use data that may leave some uncertainty about their safety. And in the traditional paradigm, we just don't have the tools to tackle this. We're really limited to those animal toxicity studies, which, as Dan talked about in the beginning, are really problematic in terms of the resources and time they take to do, questionable relevance and challenges that come in terms of extrapolating that in terms of the problems that come with clinical specifically. And having just a one-size tool, really, it's inefficient and it's challenging, and it leads to a lot of problems. And where we want to get to is something that looks much more like this. Again, this is not to be taken literally. This slide is just an example to show that we still have botanical number 1, which has enough history of use data to support its safety. But these other botanicals, and especially noting botanical number 2 and botanical number 3, which may have a lot of history of use data, but just need a couple of targeted gaps filled, maybe from an analytical standpoint or from a gene tox standpoint. And again, this is going to be weight of evidence. This is going to be specific to each botanical, but this is a far more efficient approach that allows for a much greater avoidance of those animal toxicity studies come into play. Not that they would never be needed, because we don't exclude that, but again, the mission and focus of the Botanical Safety Consortium is on gap filling with these really pragmatic and targeted in vitro and in silico options. A little spinny spin there. That is just another item just to say that this is not something that I want you to take literally, but just as sort of an example of the principle. Okay, so let's get in a little bit now to the technical working groups and the current status. And I'm really happy and excited to say that there has been tremendous progress over just the past 3 months. As the consortium has started, these working groups kicked off. Experts have been identified, and they're really getting going. And again, a nice balance of industry, government, and academic scientists. Global representation, it's exciting, it's happening. And this is where really the working groups are getting going and starting on the science, identifying the state of the science, identifying the opportunity areas within their working group. We'll start from the beginning, and of course, as we look at the strategy, the chemical analysis working group, this is probably sort of the gateway to all the other working groups, right? We want to make sure that we have botanicals that are well characterized to the level that's required for us to draw firm conclusions about how they're going to operate and work and give results within the other assays that they may be put through. So the mission is really to develop a strategy and methodologies to characterize botanical ingredients with the purpose of enabling safety assessment. So some objectives for the chemical analysis working group: again, prioritizing selected candidates for comprehensive characterization based on the needs of the other technical working groups, and looking at strategies. I think this is a really important one, to compile existing literature on analytical methods used in chemical compositions that are present in the selected botanical ingredients. You know, with botanicals, we're never starting from ground zero. There's always some amount of information out there. In some cases, there's a lot of information out there for individual botanicals. We want to make sure we leverage that as we move forward. And then selecting resource-efficient analytical approaches to comprehensively characterize botanical ingredients with respect to safety. So that last part I think is really important, because as it's been brought up by some really good questions over the course of the meeting today, and as many people know, we can't know every single constituent that's present in a botanical ingredient. They're simply too complex. But what we need to do is find those that are important from the safety standpoint. Try to isolate those, try to look at those, understand how much of them may be present within different botanicals, and then move forward in a pragmatic way to understand safety and make sure that we stay pragmatic, understanding we can't know everything, but we also have to know something, and hopefully the most critical aspects of the constituents in a botanical that relate to safety. These are the current members of the chemical analysis working group. You can see that, again, it's a great mix of folks, some really outstanding experts here, great people. As I've sat in on a meeting with the group, I'm just really impressed with how they work together, the ideas that have come forward. This is really going to be, I think, a critical group to drive consortium forward. So moving on now to the genotoxicity working group. Again, the mission, and it's great that all of these working groups have come up with mission statements to kind of focus their work in the right areas. But they're really looking for a screening strategy to reliably identify potentially genotoxic botanical ingredients. And again, applying and evaluating these results and associating them with human health risks. So minimize something there. So really, the genotox working group, again, this is another very dynamic group of folks that have fantastic ideas, incredible group of experts, and they're really looking through right now the in silico and in vitro tools that are out there to identify genotoxic agents. And for genotoxicity, some of the tools are very well established already, but not necessarily for botanicals. They've been used for botanicals, but with varying degrees of success. And again, it's that complex mixture component that makes it challenging. So really the goal here is to find those methods that can reliably identify genotoxic agents, recommend criteria for identifying significant genotoxic hazards, identifying candidate botanical ingredients, and then using case studies to look at this approach and kind of validate the approach over time. I think that the other thing that I've heard this group talk about is the importance of putting in vitro results in the context of human risk, perhaps better than has been done in the past. Really looking at dose, really looking at pharmacodynamics and how an in vitro result may actually relate to an in vivo result. I think that's an area where the group's identified challenges and would like to come up with some better solutions and some better paradigms when it comes to botanicals. This is the working group membership as it stands right now. Another group of outstanding folks. It's been a pleasure to listen to them work and begin to formulate ideas as well. Moving on to the hepatotoxicity working group. And again, hepatotoxicity is an area I think that most people think of as an important area of toxicity when it comes to botanicals, something that folks worry about. So really here, where the hepatotoxicity working group mission is to develop, again, a screening strategy that can reliably identify hepatotoxic botanical ingredients, inform mechanisms of toxicity, and characterize the, and I love this word, botanicokinetic properties of botanical ingredients. Botanicokinetic, I think, it's a great dynamic word that gets at the idea that we need to know constituents in botanicals and their kinetic properties and their absorption and their bioavailability to really help get at mechanism of action within hepatotoxicity. But it's more than just a single molecule. It's going to be multiple molecules. It's going to be a certain suite of components within a botanical that can be looked at a given time, and zoning in on those is going to be really important. So again, you're probably noticing a pattern in terms of how the working groups are approaching their issues, right? It's, again, selecting these resource efficient. I love that word, resource efficient, because ultimately our tools have to be something that can be used more widely. I think that the working groups have understood that we have to be pragmatic, that we have to find tools that can be widely used. So selecting those resource efficient in silico and in vitro tools that accommodate complex mixtures represented by botanical ingredients. And also then looking at ADME, which we had a little conversation about that earlier as well. ADME is going to be really important. It connects to that kind of botanicokinetic property that the group is coined that phrase, which is, I think, a really outstanding phrase. Selecting candidate botanical ingredients with respect to hepatotoxicity, evaluating the potential and limitations of the tools. Of course, we know that every tool has its pros and its cons, has its advantages and its limitations, and that's something that the working groups will speak to over time as well. This group is also interested in exploring the potential of drug botanical interaction. It is an important area that shedding more light on, I think, is something that will be of interest to a lot of different folks. Here's the working group members. Amy Rowe is a co-chair of this group, along with Steve Ferguson. And this is a fun group. They've had a lot of good conversations. They're really getting going. Just a very dynamic, phenomenal group of people. It's been really enjoyable to listen to them. I've learned a lot personally by listening to some of the conversations of this group. Also, I just want to mention that ADME group. Again, this is one of those subgroups that we talked about. A breakout group from the hepatotox group is sort of an identification that we have to focus on this area. This will be likely kind of a cross-functional group that will work with all technical working groups to obtain knowledge on the properties and the ADME characteristics of botanical constituents. And also this group may consider metabolites as well, which we know may be important for looking at toxicity. Okay, the DART working group. So again, you see sort of the same language in terms of how this working group is moving forward, developing screening strategies to identify potentially developmentally or reproductively toxic botanical ingredients. Same sort of strategy as the other groups. They're going to be looking for tools as well, looking at the state of the art and seeing if they can apply these tools to botanicals, which are essentially complex mixtures. Selecting candidate botanical ingredients based on suspected toxicity risk or safety as it relates to DART endpoints. And then again, this idea of establishing DART botanical case studies to really understand better the usefulness of the toolbox that they ultimately create. So that's kind of the refinement stage once they get to that. And this is a list of the working group members in the DART working group as well. I've said it about the other working groups, and I'll say it about this one as well. I mean, I've learned a lot personally just listening to some of the conversations of this group. Really, really great experts, a very diverse group of folks with some, I think, very dynamic ideas about how to move this area forward. Okay, and then a systemic toxicity working group. And this is actually a working group that I co-chair along with Nisha Sipes. And really, we have a mission to develop tools that can reliably identify botanical ingredients with potential to induce adverse effects within multi-compartmental biological systems. So there's a question earlier about, hey, how do we do ADME? How do we do kinetics and understand that sort of thing with botanicals? It's very difficult. Can we do that in vitro? And really the answer is we believe we can. There are some really incredible tools out there now, in vitro and silico approaches that enable this sort of multi-compartmental study. And the best part about it is many of these also can be pragmatic and actually cost a reasonable amount of money. And it really enables you to go and do some incredible work when it comes to that ADME function, and also in the area of in vitro and vivo extrapolation. So we have some objectives within the systemic toxicity working group we've identified, and we believe that systemic can also mean holistic, right? It's a little bit of a different connotation, but we want to make sure that we focus on that literature review process, strategies, and tools, and can we assist? Because we've had a few questions that have come up with, how do you get that history of use data? How do you gather that? And I think that doing that is a really important area that I think the Botanical Safety Consortium can make some great progress on. So we're real interested in trying to lead an effort to develop a better process for that and create some strategies and some tools around that. And then the next mission then, of course, is once you have that history of use data, can we leverage some of these multi-compartmental in vitro models to generate what we're calling systemic safety data for botanicals, right? Systemic being the idea of exposure at one point and perhaps an effect at another, which has typically been reserved for in vivo models, but technology has progressed to the degree that there are now in vitro models that can reliably look at systemic toxicity data. This is really important when we're looking at the effects of a botanical, and also considering that what's the role of the digestive tract? We take a botanical, it goes through our system, and our digestive tract, right, acts as a sort of screen before it gets into the rest of our body. And that can be a challenge with certain in vitro tools, right? We're not accounting for what gets absorbed, what happens to the digestive tract. And these models can actually get at that a little bit better and make for much better in vitro extrapolations. And that's another important aspect of this group, is using toxicokinetic modeling to support these assessments and margin of safety calculation. And really, this is getting at, again, what we try to do with animal toxicity studies is come up with NOALs, and then we apply a margin of safety to come up with a safe dose. And we like to get to the point when we leverage history of use data and then specific gap filling with botanicals that we can do this as well. And I think that's it's ambitious, but I do think that we're at a point technologically that can happen. So I'm really excited about that. The current members in this group, the Systemic Toxicity Working Group, again, myself and Nisha from NIEHS are co-chairing this. Stephen Dentali, Michael Lawless, Amy Lumen, Jim McKim from IonTox, and we've got some other folks that have joined as well, Sybil Swift from NPA. Really excited about, again, the membership in this group and what we can do going forward. And finally, the Data Analysis Working Group. And this is, again, one of those kind of overarching working groups where all the other working groups can leverage the folks within this working group to help analyze their data and come up with strategies around that. So the mission is to develop and apply data analytic methods that use in vitro and in silico data for safety hazard assessment of botanical ingredients. And this is a collaborative group. We'll be working with the other technical working groups to help refine and design experiments, supporting data analysis needs of the other technical working groups. Obviously, data analysis is a very resource-intensive task, and to have this working group available to help with that task will be, I think, incredibly important to drive things forward. But beyond that, this group will also be involved in the development, evaluation, and even the application of data analysis methods for the determination of botanical sufficient similarity. I think that's a fantastic project for this group. Sufficient similarity, which was discussed earlier, I think is really important for kind of leveraging history of use data in an effective, scientific, and valid way when it comes to safety, to understand how one raw material relates to another raw material and how different they are and what conclusions can be drawn from that. So I think that'll be just a really important project. And then again, for a subset of botanical ingredients, characterizing the degree of similarity between the toxicological profiles derived from in vitro and in silico data compared to those obtained from traditional animal tests. The current membership in the data analysis working group is here, and again, this is it's great to have some of the CROs present as well. I think that's excellent that Eurofin has joined. It's important that we have that sort of expertise as well, that analytical expertise within this. We've got academic folks. We've got just a great group of people here to drive this work forward. Okay, so just a couple of words on the sort of botanical candidate criteria and how we're picking these, right? So how are the working groups contributing to this process? Each toxicity endpoint working group will select probably in the neighborhood of 6 to 8 ingredient candidates. And this selection is going to be based on evidence of toxicity or safety from scientific findings, humans, animals, or alternative methods. And I think the idea here is to pick botanicals that you think might be positive and botanicals that you think might be negative. And once these lists are created, they'll all be compiled into a master list and then undergo chemical analysis. And then this master list of botanical ingredients can be tested through the different working groups, and then these results will be made public. Just some considerations on methodological criteria. I think a lot of this has been touched on. Our focus is on the in vitro and in silico side. Again, it's establishing existing methods that are out there for botanicals, which are complex mixtures. We want to make sure that we leverage history of use data for individual botanicals. That's really, really important. We're not starting from ground zero with botanicals ever. In vitro and to in vivo extrapolation to build confidence in what you can take away from these in vitro tools is going to be a really critical aspect to methods that are considered. And finally, practicality. Again, as I mentioned, it has to be cost and time effective while still meeting the technical requirements. It's got to be something that can be widely used. That's really what some of our criteria for the methods that we're looking at. Also, something that has already been hinted on as well is that there's certainly the possibility for other groups. Really, the sky is the limit here. We're gonna listen to our stakeholders, and we're gonna understand where the need and is for these different groups, and work to fill them as we see that need. Cardiotoxicity is probably highest on the list right now, but there's other possibilities: immunotoxicity, nephrotoxicity, neurotoxicity, and of course the list can go on. And again, we don't want to stretch ourselves too thin, but we want to make sure that we tackle important areas within the Botanical Safety Consortium. We have a nice wide viewpoint of all the different safety concerns that can come out of botanicals and be able to address them in an appropriate, pragmatic way. So to summarize, the VSC has established 6 technical working groups to date, and there may be more to come, to execute the mission and objectives of the organization. I think that it's a good moment to really thank the folks that have joined these working groups. It takes dedication, time, expertise, and really determination to be a part of these working groups, to drive forward strategy, to sort through the details and make decisions about how to move forward. And again, it's been a real pleasure, a real learning experience for me to listen in on the conversations with the experts that we've been able to bring together so far. And because I compared all the working group members to the Avengers, I did have to bring that back home a little bit. And I think this little conversation between Captain America and Iron Man sums it up pretty well. Captain America, we need a plan of attack. And Iron Man, I have a plan, attack. And I like that because I do think we have an organization, we have a structure that has set us up for success. But I also think that with the technicals and all the different issues that come in, you just got to dive in and you got to start sorting through it. And that's what I've been really impressed with the groups, is the willingness to dive in areas that may not have a complete amount of certainty and start attacking these issues and figuring out how we can come up with a strategy and from the safety standpoint, move things forward. So that's really my show, and my slideshow is done, and I'll pass it back to Michelle.
Thank you so much, Joe. Appreciate it. So I just have a few questions before we move on to Stefan, who's going to talk a bit more in depth about some of the chemical analysis piece. I did want to mention, and Joe did put up the names of the folks on the working groups now, but we are actively looking to identify additional key stakeholders, as I think he mentioned and Connie mentioned. So please know that we are still looking to add additional people to help us in the technical efforts. So there were 2 questions from Jiang Hu related to the BSC being open to having new technical working groups down the road. I think Joe touched on some of the ones that have come up in Jim's discussion. That's going to come up as well. I think we're very open to additional technical working groups, and the process would be to get in touch with us and we can scope things out, determine what the needs are, who the key players are, and how it fits in with the objectives of the consortium. On that same note, and this has come up a couple times, and I will open it up to either Joe or other folks on the steering committee, a question was whether there will be a dedicated technical working group for botanical exposure assessment, since of course exposure is the other key element of risk assessment. And I was wondering if anyone would maybe speak to that from those who have been involved in the initial discussions on the consortium.
Well, I can take a first stab at it. All of the working groups are absolutely considering exposure assessments within their overall strategies. I've heard it talked about in all the working groups that when we look at these methods, they have to be put in the right context later on. Incredibly important. It's a great point. At this point, I don't know if there's going to be a dedicated working group to that. It's certainly possible. Further discussions probably need to be had, or if it's going to be handled sort of within the working groups themselves. But ultimately, you're right. It all has to be put into context. That exposure risk assessment is a critical piece. And at this point, it certainly is on the radar. And that's what I would say at this point.
Mm-hmm. And I think just like that ADME group, if we decided to have a group that is specifically focused, maybe from key members from each of the working groups to come together to have one that's specifically addressing the exposure piece, we can do that as well. A lot of what we're trying to do within the consortium is be flexible to make sure that we can form the appropriate groups as needed. Just one other question is related to involvement of clinicians. And I think that if there are clinicians who have a vested interest in trying to address some of the safety related to this, we are open to having them on the group. So it's really just a matter of getting the word out and getting the right people plugged in. I don't know if anyone has any other comments on that.
Well, I think it's incredibly important to try to get clinicians involved in the Botanical Safety Consortium. I mean, that's a point of view that is really important to the overall puzzle, if you will. And I would absolutely hope that clinicians would find a place in the consortium, be able to join and provide their perspective, because it's a very important one.
Okay. So there were a couple other questions. I think we're going to save them to the end because they kind of wrap into a lot of this discussion. So we're a bit behind, but I'm going to pivot to Stefan Gafner, who's going to talk more specifically about the ingredient characterization work.
Thank you, Michelle. Thank you, Michelle. It is a great pleasure to take you down the road into the fascinating area of plant metabolism and on how to analyze them. I'll explain the cheesy part of my title a little bit later. So let's get into the slides. I just want to talk briefly about the American Botanical Council. For those who are not familiar with ABC, we're a nonprofit educational organization that provides information about the responsible and safe use of medicinal herbs. We have a fairly wide array of members, mostly just people that are interested in medicinal plants, but we also have support from healthcare professionals, researchers, educators in the industry, and we've been around for over 30 years now. Next slide is Chemical Analysis Technical Working Group. You've seen that from Joe's presentation. The mission is to develop a strategy and methodologies to characterize botanical ingredients. We do that by prioritizing selected candidate materials, do literature search, and then propose resource-efficient analytical approaches. And as Joe pointed out, we have a great group of members of the Chemical Analysis Technical Working Group. Some of the people I've worked in the past, and I'm just really thrilled about all the expertise that we have in this working group. Now, the cheesy part. I selected the title. You may be familiar with the history or the story of Scheherazade, who volunteered to get married to a sultan despite his habit of killing his wife after the first night because he was worried she would be unfaithful in future. So in order to avoid being decapitated, Scheherazade started telling a story to the Sultan, and then just before the story ended, usually on a cliffhanger, she stopped talking, and so the Sultan couldn't execute her because he wanted to know how the story finished. So the next day, Scheherazade would finish the story and then started a new story, ending again on a cliffhanger, with the idea to basically telling 1,000 stories for 1,001 nights to save herself from harm. And the parallel to the Botanical Safety Consortium and the Chemical Analysis Technical Working Group is that we will analyze 1,001 compounds or more to save our consumers from harm. So I just want to put in one slide about the beauty of plant chemistry. I'm always fascinated to see what kind of compounds plants can come up with. Here are 4 different structures, and I have to warn you, for those who don't like chemical structures a lot, they may have the opportunity now to go and have a coffee, because there will be a few more that I will show during my presentation. So we have here azadirachtin, which is a nortetraterpenoid. That's an anti-insect repellent compound from the neem tree. We have methylbuvardine, which is a cyclic peptide that has anti-cancer properties. Then on the right-hand side, vincristine, which some of you may be familiar, which is an alkaloid from the Madagascar periwinkle, also with anti-cancer effects. And then we have a phenolic, highly colored compound, hypericin, that's found in St. John's wort, and it's a naphthodiantron structure. So plant chemistry can not only be beautiful, but it can also be challenging. There are estimates that between 500 and 10,000 different molecules occur in a single plant species. My personal thinking is that it's probably more towards the 10,000 than the 500, given the fact that there are already over 500 compounds that have been isolated, for example, from ginger. Broadly, people distinguish between primary metabolites and secondary metabolites. The primary are essential to the survival of the plant, are directly involved in growth and development. Examples would be the DNA in plants, proteins, starch, or cellulose. And then the secondary metabolites, they're facilitating the primary metabolism. They allow adaptation to and survival in the environment. They're often involved in the defense and reproductive mechanisms. Examples would be terpenes, alkaloids, or flavonoids. And obviously the line between the two are blurred because some of the primary metabolisms are also involved in some of the effects there. So here, that's other slide on the challenges and the factors that impact the ingredient metabolome. Dan has laid the background very well. So we have climate that impacts, cultivar can impact, agricultural conditions, so fertilizers, compost that you add, but also a lot is due to the soil and the microbes that inhabit the soil, like fungi and bacteria. The same above ground, you have fungi that can impact the metabolome, then obviously processing and stability. And I go a little bit more into detail about processing steps. Here, the 4 buckets I talked in the previous slide, the importance about raw material. So all the factors can impact the raw material. Then once you have the raw material, if you do an extraction, the solvent will make a big difference. Processing equipment can make a difference in the chemistry, and obviously the type of process, type of extraction, temperature, duration, pressure, that will impact the ingredient chemistry. So at the end, you may have a wide variety of ingredients made with the same starting material. One example, just to show the differences in metabolome. When I was working in the industry, we had an initiative looking at potential adulterants for skullcap, that's Scutellaria latriflora. One of this is Mars skullcap. We got 3 samples, so 2 are botanically authenticated, one from the province of Quebec, one from Oregon, and a commercial sample. And if you look at the material from Quebec and Oregon, you will see that just based on the phenolic composition, these are 2 totally different metabolomes that you find in basically the same plant species. One slide on how the solvents may impact the chemistry. Here on licorice, we did a comparison between hot water and some of the more lipophilic solvents, and you see again that depending on the solvent you use, you get a totally different composition in your final product. So that's a really important consideration if you work with a plant, for example, like licorice. And there are a number of additional challenges that are sometimes with regard to agricultural, it's going a little bit too fast, agricultural conditions, so heavy metals, pesticides, herbicides, microbial contamination, could be environmental, could be processing, but then also we have more recently through processing polyaromatic hydrocarbons that have been raised as an issue. Other contaminants could be from storage containers, and then adulteration. And I've been asked to say one or two words about the Botanical Adulterants Prevention Program, which ABC is involved with our partners at the American Herbal Pharmacopoeia and the National Center for Natural Products Research at the University of Mississippi. And this is a program to educate herbal and dietary supplement industry members about ingredient and product adulteration. The program has been around for about 10 years now, and we've published a number of reviews on particular ingredients that just gives you a sort of a snapshot where we are. We've published 20 bulletins, so short reviews on ingredients where adulteration is occurring presently in the marketplace, and then there are a few that are about to be written in edits or that we have planned. So these bulletins and other products are all available free of charge through the ABC website if you're interested in that. Now, I have to say there are almost as many analytical methods as there are secondary metabolites in plants. This is a slide from a yet to be published review on analytical methods for botanical ingredients. You will see the most widely used approaches on the left. I highlighted the ones in yellow that I believe that industry currently is most frequently using, and you see that some can be used on the whole plant, some just for extracts, and I also have a number of limitations. So each method has to evaluate it if it's fit for the intended purpose, and not any single method can do everything. When I usually start talking about analytical methods, I start with botanical taxonomy, because that's still the starting point for how we establish the nomenclature of plants. It's not as easy as people may think. I have an example of eyebright, which is a species that hybridizes frequently. It can have different ways of pollination that leads to different morphology. So in case of Euphrasia minima, you can have yellow, purple, or white flowers. And then depending on the botanist or the taxonomic database, some put the same species in one group, some split them up. So that leads to a situation where the botanists often use different scientific names for the same species, which makes the correct identification quite challenging. We have done an investigation of commercial eyebright samples that's still not published. There was a collaboration with a number of parties, including KMAG, the HPLTC manufacturing company. We've collected 28 botanically authenticated samples, 32 commercial samples, so mostly bulk materials that came from the Eastern Europe, and 7 finished products. These were all from the United States. And just put that in to show you the variability in these products. So that's what the commercial eyebright product look like. So just from the fingerprint, the HPTLC fingerprint, you see that the eyebright is not equal eyebright. And in addition, you have adulteration going on. So of the bulk materials, 8 were adulterated with odontitis. And you also have some of the materials, bulk and finished products that are basically show you a blank HPTLC tray. So these are on the lower left where you really don't know what the manufacturer or the supplier used. And that for me is another area of concern. So this leads me in a next topic. Still working on identification. We'll get into microscopy, and I have a case here of skullcap and germander. In the 90s, it was known that germander species are used to adulterate skullcap. A problem that is of toxicological relevance because germander species contain neoclerodon diterpenes with oxidized furan ring. And so these are known liver toxic compounds. The mechanism has been established, so it is really important to keep the germander out of the marketplace. And that slide shows you just a simple comparison of photomicroscopy images. These were provided by our friends from Alchemist Labs. You see that with the skullcap, you have different type of trichomes or hairs. In Canada germander, if you can see my arrow, you have a lot of these long hairs. With wall germander, you have these club shade hairs, and then you also have the interior of the stem that's filled with cells, whereas in skullcap it's hollow. So you can distinguish them fairly readily by microscope. Another interesting plant that I've worked with quite a bit is licorice, and there's actually a museum devoted to licorice that I always wanted to visit. So if anybody ever gets into the area of Rossana in Italy, I highly recommend to go and check it out. So now we're getting into chemical characterization. So in my days in the industry, we worked with a supercritical extract for which we didn't really know what the composition was. So we did LCUVMS, and then we noticed that we need more work. So we did it the old-fashioned way, basically isolating each of the compounds that we could. And you can see that the metabolism is very different. On top you have, and I have to go back, coumarins, then you have isoflavones, and then on the next slide, apologize for all the chemical structures, isoflavones, pterocarbones, aurons, and simple phenolics. Some of these have the same molecular weight or isomers, so they're not that easy distinguished just based on online method. The advantage of that type of approach to characterize a botanical ingredient is that you can get samples of the isolates and to do additional testing. The disadvantage is that it takes a lot of time, and mostly industry and doesn't necessarily have the time to go through all the tedious process of isolating these and do NMR to characterize the structure. So there are people that have been very successful at characterizing ingredients online. This is a very elegant approach published by Vincent Sika and his colleagues from Procter & Gamble using HPLC with UV and charged aerosol detector, also coupled to HRMRS, so they have a split valve. Some gets into this CAD and some in the high resolution MS. With that approach, they've been able to determine that the composition was about 75% of proanthocyanidins with a higher molecular weight or polymerization degree above 5, 7% 5 and below, then a little bit of lignans, and the rest minerals, carbohydrates, and a few lipophilic compounds. And scientists used quantitative data to look at the compounds that could be possibly needed for further evaluation. Those that were above the threshold of toxicological concern, which is indicated here, would be evaluated further in further literature and possibly bioassay work. So that's one of the approaches that we definitely consider for the Botanical Safety Consortium. Another way to characterize a material very thoroughly has been published by colleagues from Herbalife. And in this case, the scientists used 8 different official methods to calculate the composition or determine composition, ended up with about 95% of what was in that aloe extract. And so another way to get to the composition very elegantly. Now, as it wasn't complicated enough just to determine the composition of the extract, one question we have to ask: what happens if we put our extract or our ingredient into a test solution if we do an in vitro bioassay? So when I was in the industry, we usually had stress tests as part of method validation that would include exposing the test ingredient to hydrochloric acid, sodium hydroxide, and peroxide. And you see here with liquor isoflavone A, after 22 hours in alkaline conditions, you see a degradation product popping up. That doesn't happen only in alkaline condition. It also happens if it's in methanol or ethanol water mixture. It's a little bit slower, so with alkaline conditions, you can accelerate the degradation. We were fortunate enough with a collaboration with Procter to identify the degradation product as an isoflavone quinone. Here I have the proposed reaction, so it's an oxidation, probably mediated by catalyst, a metal catalyst. But what I wanted to show you here is that in this case, the antibacterial activity that we were interested in when we compared the original compound liquor isoflavone A and the degradation product, there was a over 10-fold difference. So that can play a huge role in a toxicity assessment as well if you have a product that is rapidly degrading. So that's another area that we have to investigate as part of the chemical analysis technical working group. So here are the method considerations of the working group. We'll have to look at the availability of reference materials, look for methods that can see inert material. Then we'll have to be choosing methods that we can adapt for mixtures, have to investigate sample preparation issues, stability issues, and detection bias. That's a particularity to depending on the type of analytical approach that we'll use. It tends to favor some of the components over others depending on the detector that you're using. And this is the approach. I think you've heard some of it. We start with literature compilation. We'll have to figure out where we get the raw materials and finished products from, have to authenticate the material. Then we'll do a comprehensive characterization. That includes contaminant analysis, and we also will include bulk stability. What we won't include is right now isolation and looking for the actives. I think if that's warranted, that can be discussed, but at the moment we really want to focus on the botanical ingredient in its complex mixture in its entirety, rather than starting to isolate and get fractions and retest, because that's just a little bit out of the scope right now. And with that, I just want to thank everybody that helped me gather this data over the years, and I'll hand the microphone back to Michelle.
Thank you, Stefan. All right, so there's a couple questions that have come in, but I wanted to specifically see if anyone had any clarification questions from Stefan's talks about the chemical analysis part of this. There's one that came through related to, in regards to the botanical selection, where there'll be input or data drawn from manufacturers. So traditional patent medicines or Western products that are more clinically targeting with higher potential for harm. And the examples given were milk thistle and silymarin used for liver support that may be contaminated with hexane.
Definitely to the first question, the answer is yes. Yes, we would welcome input from the industry on materials that BSC should evaluate. And as you've seen in the slide, the contaminant analysis is part of what we're going to do. So if we'll have a mixture of milk thistle that has a substantial amount of hexane, we would obviously write that up. But I don't necessarily think that such a material would be representative of the marketplace, because there are limits of residual solvents, and if a ingredient is beyond those limits, I mean, we can say, okay, there's an issue with the residual solvent, but it doesn't mean necessarily that the botanical itself is a liver toxic ingredient.
Thanks, Stefan. And there was a question from earlier that I had flagged for Stefan related to any experience on the impact. In this case, it was specifically on climate change, but different agricultural practices and the impact that that might have on the chemical composition within plants and how that would parlay into botanicals.
I love questions about sustainability and the impact of sustainable agricultural processes on the botanicals. On one hand, we definitely want to have a wide array of materials. So if there is a plant where there are enough samples grown from sustainable areas or sustainably, and those that may be harvested or grown under unsustainable conditions, that's something that I think we can look into. But on the overall premises, I would say sustainability is not a focus of the BSc. We really want to have a focus on the safety, but as I said, if there is a reason why we should look at sustainable versus non-sustainable harvesting practices or growing practices, I think that can be discussed on a case-by-case basis.
Great, thank you. So I think we'll move on to Jim, who's going to kind of wrap us up a bit, and then we can spend some more time on additional questions. And there's a few other ones that have come in that I will bring up at the end. So Jim, are you able to share your screen, or would you like me to pull up the slides?
Could you pull those up, please? Only
yes.
Because the Mac is going to be a problem.
Oh, right. Now I'm remembering now. I forgot. Okay.
That's okay.
No problem. Just give me 1 second.
Yeah, there's always a problem when you're trying to do these Mac to PC to Mac to PC presentations, so I apologize.
Yeah, no worries. No worries. Okay.
There you go. Now I'm going to take control at my end on the pointer.
Yeah, give me 1 second. Let me just do that. There you go. Okay.
All right, thanks. Thank you, Michelle. It's been a long day, so it's nice to be at the back end of today's informational unveiling of the BSc. And it's also nice to bookend today's event with my colleague, Dan Marsman, as we were both at the, I guess, the moment of conception, or at least one of the most visible starting points, which was the 2017 Eurotox meeting. So I want to be a little bit redundant, and let me see if this slide will, a little bit redundant, but per Dale Carnegie, you got to tell the audience what you're going to say, and I thank Dan and Connie. Then you're going to say it. So thanks to Cynthia, I guess Cara, Joe, and Stefan, and then tell them what you've said, which is me. So these slides I know are starting to look very, very familiar. So I'm just going to recap this with one word each. The first one is engage. The second one is to characterize. The third one is identify. The fourth one is evaluate. And the fifth one is integrate. So 5 easy goals, what the BSC aims to achieve within a multi-year time frame, I guess with the available resources. And the dozen or so strategic elements to accomplish these grandiose plans. You've also seen this slide before. I'm not going to spend much time other than to just point out that what everybody has said so far, developing a list of candidate materials, and these would be materials that already have some kind of safety data. You don't want to be focused on de novo botanicals because you have nothing to compare to. Of course, the reference library, all the information on characterization, identifying the suitable assays, getting this mega library in place, analyzing and reporting. And I think it's important there to point out making the data publicly available. And then finally, all of this should end up as a framework or toolbox. Here's a really nice presentation of a flow chart. For time's sake, I'm not going to dwell on this other than to say that there's many things happening in parallel and not sequential. So I think that, once again, is an important point, that as the various TWGs come up with their priority endpoints and do the searches, there will be a library developed that all of the working groups will have access to. And then as botanical candidates are identified and as assays and approaches are identified, these are all happening within those TWGs. And eventually there will be the reporting and that framework development. So this is the ongoing stakeholder communication and engagement, and that's where hopefully those that are stuck around to the tail end of this will have a chance to acknowledge or at least participate if interested. These slides you've seen as well. This is, of course, my slide on engage. And once again, I think it's important to mention the global stakeholders, and hopefully I get some sense that there are some of the internationals on this particular BSC webinar, and I apologize that the timing, at least chronologically, may not be the best, but hopefully you're still engaged in whatever time zone you're located. But we want that global participation. We want various academics, government, industry, consultancies, trade association from where I come, etc. We want it to be a scientific endeavor that will have a regulatory application. And there's areas where you can engage. You saw that, I guess it used to be an onion, but now it's a leaf that has those various layers of engagement with the BSC, whether it's the stakeholder, the working group, or even eventually in a leadership role. And I think it's important to point out that this will go both ways. It'll be communication and activities from the BSC, as well as getting that expertise coming into the BSC. And so I think the take-home question at this particular slide is, how can we expand and enhance our network? We had dreams of going to Eurotox again this year and other international meetings, but 2020 will be a year to remember, and unfortunately it will curtail many of our physical opportunities to go international. The next slide was my characterization. I think you heard from Stefan and others that there are tens of thousands of constituents, and so there needs to be a way to come up with the appropriate levels of which chemicals are important, using targeted, non-targeted approaches, and of course develop strategies. And so the takeaway question, what are some of the key challenges regarding chemical characterization that you have experienced? And so we would love to hear from you and have you participate either through email input or, more importantly, filling out that form to demonstrate even more engagement. Assays and approaches. Once again, I think my keyword was identify. I like the word pragmatic, which is we're looking for assays that are sensible and realistic, I think based on practical rather than theoretical or idealistic considerations. So once again, I think pragmatic, fit for purpose assays are important. What approaches are within the domain of applicability? And do we need to modify anything to take into account the fact that we're dealing with botanicals and not other single moieties? And are there examples of assays that may be suitable for the BSC to consider? Analysis and application, I had said evaluate. So this is compare the new way with the old way. We can only do this if we have a Rosetta Stone. I think if we look at how it has been done historically, and run those same types of materials through new batteries to see what the concordance between those assays might be, then I think we have an opportunity to slowly replace some of that gold standard with a new platinum standard, as it were. I think that the 20,000 plus botanicals that Dan mentioned at the beginning of this presentation would have an opportunity to be characterized in some fashion. And so the takeaway questions are, what are some of the key challenges regarding data analysis and interpretation that must be considered? And what types of data sharing formats could be the most useful for the BSc to consider? And finally, this is all for naught if it's only a scientific exercise. We need a widely accepted framework that scientists and regulators and public health can rely upon. We want to leverage those learnings. We want to identify the challenges, if there are any. Of course, there's challenges, and prioritize research, look for those gaps, and maybe explore improving and new analytical methods that come into vogue. Takeaway question, do you have some ideas of how the BSc might demonstrate the utility of this framework? And finally, let me move to this slide, which also was shown earlier in today's presentations. And once again, the priority endpoints, the ones that have already been chosen, and you've heard about the cardiotox, hepatotox, systemic tox, I guess genotox, and DART. There they are, and also scoping out what other ones might be worth exploring. And so once again, our question to everyone is, what other endpoints should be considered as high priority, with the understanding that we would definitely need to have some type of ability to make sure that they were worth exploring and putting the energies into. And finally, hopefully all that you've heard today resonates with your needs as a botanical safety scientist, as a concerned public health official, confronted with an ever-expanding plethora of new and unknown botanical products scooped up by the population, maybe an integrative or naturopathic doctor that refers or cautions their patients on new treatment modalities, or even a federal or state regulator faced with a dossier that needs a go, no-go decision to market. If yes to any of these, we hope that the time you invested into this webinar was and continues to be of value. And on behalf of everybody, I would like to thank you for participating as an audience, and we welcome any guest questions at this point. So I will turn it back to Michelle.
All right, sorry, I couldn't find my mute button. So thank you so much, Jim, for kind of teeing up some of those questions. I have some, hold on, I'm just going to take control back here. Have some questions that have come through. A couple questions related to the data for the Botanical Safety Consortium. So obviously there'll be a lot of data that are generated. One of the questions was related to the most suitable forms for making the information developed by this consortium available. I think Connie already covered, we plan to make it publicly available, and the way that we generally operate is via peer-reviewed publications, of course. So obviously there's many interim steps related to the data when they're generated and making it available. We have to sort of develop a suitable approach, but the plan would be to make it publicly available when it's published. So most journals right now require that data be made available as supplemental information, et cetera. One of the things going further than that, though, is to get feedback from our larger group of stakeholders related to, is there a need for something like a database or some, not only to make it available, but to make it easily accessible. And if you have particular thoughts on the best way to structure that and develop it, obviously we have a lot of partners in this effort who have experience in that vein. So we're open to your thoughts on once that the data are published, how to make it more easily accessible. That said, there was also a question related to, as the data are generated and before it's published, obviously the folks that are involved in the consortium on the technical working groups and the stakeholder council who are members of the consortium would be able to see those data. Obviously it's a group effort, and we would make it very transparent and available to the folks involved. And again, the point is to make it public through publications. Does anyone else on the steering team have anything in that vein related to the data availability, public availability of the data that they'd like to bring up? No, sounds like no. Okay. There's a question related to the eventual Library of Botanicals. Will that library have corresponding toxicity and safety data on multiple preparations and extract types for the same botanical source? So, i.e., will the data generated facilitate that read across or bridging between different extracts of the same botanical? Would anyone like to speak to that?
This is Cynthia. I just wanted to step back 1 second on the data availability and mention that we do have publicly available databases that could be taken advantage of to house the data. So that's one option that we can discuss in the future.
That's a great point. We don't want to reinvent the wheel if there's already a great system out there. So definitely leveraging.
This is Joe. I would respond to that by saying that, you know, you really have two choices when it comes to botanicals. You can go horizontal or you can go vertical, really. You know, you can go wide and do a lot of different species, or you can go more vertical and try to, you know, do different types of extracts. And honestly, I think we have to do both. I think that resource constraints and our strategy, and also, you know, what our stakeholders are telling us they're most interested in, I think that's going to drive you know, where we go with that. I mean, I think the answer is it would be great to select certain botanicals and try different types of extracts, because I think that's a really important issue. But we'll also probably not do that for all botanicals. So I think that's maybe the answer to the question.
Yeah, it's a good point. The other part of this is that we have to start somewhere, and I think there's all sorts of really great ideas of different routes of exposure, dermal, for example, that have come into play, inhalation, additional types of questions related to other working groups and things like that, other endpoints. So I think that if this program grows and we get additional ideas once we get sort of this initial scope of work developed, that's definitely something to explore. But we also are very cognizant of making sure that we don't make our remit so broad and so overarching that we sort of are paralyzed by all the different things that could be done. So I do think there's lots of questions that have come up, not only on this webinar today, but in others related to the breadth of botanicals on the market, the different extracts. And I think we're open to those discussions, but also want to make sure that we address the initial objectives and mission that you've heard about today. So there's another question, probably for any one of my steering committee members. With botanical supplements, oftentimes activity and safety is a, quote, composite phenomenon, and they may not have activity in isolation. For example, the toxicity of one may be nullified by another. So you have component interactions. And individual constituent characterization and activity falls in the realm of medicinal products. What is the expectation for a dietary supplement? Is clinical data on safety and efficacy at the extract level sufficient, or is the expectation at an individual constituent level? This has come up many times. Someone want to take that one on?
I feel like there may have been multiple parts to that
There
question.
were.
Yeah. Okay. So I do think we're focused definitely on the whole mixture, and use of constituents to better understand certain aspects will be limited to cases where that is necessary, to be determined, I would say, on that front. Can you repeat the first part of that question?
Yeah, it's, sorry, I just lost it. Here we go. The fact that the toxicity and the activity may not happen in isolation, right?
So I think I might answer a slightly different question, but it's relevant here. I think part of the challenge to the Botanical Safety Consortium is to better understand the threshold of when bioactivity transitions to toxic effects. And that is an important part of what we're doing, because I think a lot of times many in vitro assays are pretty sensitive. And so you can see biological activity without moving into the sphere of toxicity. So I think that's critical to our mission, is to better understand that transition from activity to adverse. And about the constituents, the whole being more than the sum of its parts, or different constituents doing different things that's exactly why we want to work with whole mixtures instead of taking it apart and reducing it to the constituents and looking at those in isolation. I think that's one of the drivers for looking at the whole mixtures.
Yeah, I'd like to add
And then
to what Cynthia, sorry, did somebody else want to say something?
No, Joe, I'll get it after you. This is Cara.
Oh, okay. I was just going to add to what Cynthia said, which is, I think it's very tempting to want to break down the botanical, right? Because the more you break it down and get to something individual, maybe it's a lot more comfortable. I think the thing about botanicals that consistently challenges scientists of all sorts is that there is always going to be an unknown fraction of that botanical. Sometimes it's a large unknown fraction, and sometimes it's a smaller unknown fraction, but you're simply not going to know everything in all the constituents in a botanical. And therefore the toxicity or efficacy that comes from botanicals, you don't always know what constituents may be acting together to produce that. And that doesn't mean that you can't go a long way in your safety assessment and come to conclusions, and really strong conclusions. You may not know exactly what's causing it, but you can still get to a pretty good place with safety. And I think that's something that we have to grapple with as stakeholders and scientists, and still make progress with botanicals.
Thanks, Joe.
And then I was just going to add on that from FDA's perspective, we're looking at what the ingredient in the product and then how the ingredient is being used in the product. So the serving size, the total conditions of use. So we're going that's the picture we're going to be looking at. But I think the consortium is designed to then, the end product is looking at a framework or a toolkit that you can then, that an individual stakeholder ideally can then take and apply to their own ingredient. So if you are looking at just a single constituent or a very refined extract, that may be different than, of course, a very crude extract. But hopefully the principles that are discussed within the consortium can then be applied to your particular ingredient and product.
Great, thanks, Cara. So there have been quite a few questions more broadly that I wanted to pose to the group on the phone here related to botanical drug interactions and botanical interactions. And I know this came up before of, the complex mixture on top of a complex mixture, but one of the questions was related to this consortium's effort to identify assays that may be suitable to evaluate that, as well as if there would be any sort of movement towards developing some database or expanding on existing databases that articulate those known interactions.
This is Cynthia. I think Amy Rowe gives a really nice talk on some of the available methods for looking at drug, botanical, and botanical interactions. And I think that is definitely on our radar and part of the Hepatotox ADME group's mission to look into those. And I think as part of making all of our data available, we hope to contribute to the greater understanding of those and their occurrence.
Thanks, Cynthia. Yeah, I think there was a question of which of the groups. I think the group that's talked the most about the drug botanical interaction is that hepatotoxin ADME group, but it'll obviously be something that comes up in some of the other ones as well to some extent. But that's the one that's probably focused on the most. There have been two questions I've seen about the use of artificial intelligence, machine learning, and the the role of that within the consortium. I know it's within our scope to help identify in silico approaches and tools. I don't know if anyone else on the group today has any additional thoughts they'd like to provide on that.
This is Joe. We've definitely, I believe, hinted at least thinking about that sort of thing, right, in the systemic toxicity working group. Modeling is going to be, and computational modeling is going to be a big component of what we do. I think that, you know, artificial intelligence it's a pretty big word, and exactly how we would apply that, I'm not exactly sure yet. But I mean, that's the sort of stuff that we want to, you know, go after. We don't want to exclude. I mean, I think great ideas around how that can be applied would be really open to receiving those. You know, I think certainly using computers and using modeling and you know, leveraging algorithms is a really critical part of how we make things more efficient for safety assessments. So that's my comments.
Thanks, Joe. So I think this question might be for Stefan. I don't know, I'm not familiar with the acronym, but it's, will IRMS-based methodologies become standard testing methods to identify biological versus synthetic adulteration?
I assume that's for me.
I think so. I have no idea what.
Yes. No, it's stable isotope ratio mass spectrometry. So it's a method that's been around for a while, just to look at for example, with turmeric, to see if there's synthetic curcumin into your in your turmeric extract. And right now, that has not been discussed. I think that's again a case-by-case basis, but it is my hope and understanding that we want to focus on authentic materials. And if we go and look at a selected market product, we may have to investigate into that, but right now it hasn't been discussed. So I really can't promise that there will be any stable isotope ratio measurements as part of the chemical characterization right now. But I think on a case-by-case basis, we need to look into that.
Great, thank you. I think I'm just walking through to make sure that we've addressed most of the questions. I think there were a lot related to prioritizing botanicals for testing. And I think that one thing to note for those of you still on is that what we're trying to do, and we've stated this, and it's sometimes difficult, but we're trying to identify assays that would be suitable. And so we're really trying to identify that strategy that was shown on a couple slides of identifying botanicals for which we have available information that would be suitable to help elucidate and determine whether the in vitro and in silico approaches are going to be useful down the road. So it's not coming up with a list of botanicals that we think need to be tested more than others. It's coming up with a list that's going to allow us to develop and identify the most suitable tools and approaches. And there's a difference there, and I think that's important going forward to keep in mind. I think those are most of the questions. There's quite a few on the list here. I think some have been covered in other places. One of the things I will commit to doing is we have these, of course, all in front of us, and I will share them with the presenters, and we will provide the answers. We'll either go back and get the answers that were provided orally and sort of do a transcript of those, and then any that maybe weren't answered as specifically, we will follow up on and make sure that we address those. So if we didn't answer your question orally, or if you had to leave the webinar, be assured that we will work our best to get to those. So one last thing is all the folks who registered for the webinar today, I will on probably Tuesday morning be sending out, because I know a lot of people probably their end of their workday is finished. We'll be sending out a short survey, monkey survey, to get your feedback from the webinar, answering asking some key questions from all of you related to additional information you'd like to know and learn about the consortium. So I would just implore you to please fill that out. It should only take a couple minutes. We tried to keep it short so that we can get feedback, keep you engaged. It'll give you a chance to ask any additional questions. And with that, I think we will adjourn. I would like to thank all of the presenters for the great work that you did and the huge list of attendees. We had a lot of people, over 300 folks on the webinar at one point today. So appreciate everybody's time sticking in front of your computers for 4 hours. And we look forward to hearing more from you and working with you, hopefully, on the consortium as we go forward. Is there anything else from the steering committee that you'd like to bring up? Okay, well then with that, thank you all. Have a great weekend, and we look forward to working with you soon. Take care.